Good.
All right. Yeah. Okay. Yeah. So I think originally when you contacted me, you wanted me to talk about more label-free interferometric type of stuff, which would be fluorescence-free. But this one is full with fluorescence. And I can then talk about the other stuff in December.
When you're
Or
here.
in November. Yeah. Right.
Just for Wahid and Shudipto, we are live on YouTube. Wahid, okay?
Okay, I won't say anything that
Should I
I
start?
No,
that's
you
just
can. I think it's fine.
just to make yourself alert. Yeah.
Yes, no, that's
Should I
good.
start, Jyotishman?
So it's 11:50. Yeah.
Just the welcome things and all that. Let me know.
Yeah, you can start, Shudipto.
Okay, so hello and good morning to everybody. So we're in the 15th edition of what we lovingly call FCS. The name originated from fluorescence correlation spectroscopy, but it has grown much, much, much beyond that. So we started in 2009. This is a workshop that happens every year. And this is the 15th edition, where we try to teach each other how to do things right, how to do things better, how to make instruments, optical instruments that has applications to either biology or to material science mostly. So this year, it has actually grown into having 4 parts. The 4 parts are, of course, the last part is the Optics Within Life Sciences Conference. It's a mid-sized international meeting, probably a large size by usually what our field sees, especially in India. And we'll have a lot of people, including today's speakers, in there in November in IIT Bombay. So that's the last part. That happens from 18th to 21st of November. And then the actual workshop part, which is from 16 to 18, will also happen in IIT Bombay. But that requires us to sort of handhold, interact directly, and sort of tell you all we know, in front of the instruments and actually building some instruments, etc. That we cannot admit everybody. So like every year, we are oversubscribed this year for the workshop part of it. I think we have only a place for 80 or so people, and we had some 300 applications. So then we thought of something that everybody doesn't miss on that part. So we have this online thing a month before. So it's like a curtain raiser for the main event. This online thing also has 2 parts. One part is really online, teaching you how to handle the instruments, talk about methods, etc. And when some of you will come to the actual workshop, you will do this actually with your own hands, and there will be more detailed lectures there. But this tomorrow and day after, the 2 days that we are having following today's, will be dedicated to an audience that may not be there. All of them may not be there. And also more basic stuff so that you don't waste our time talking about really basic things. So that's great. But what is the 4th part? 4th part is new, which is today. And this is what we can call it the frontiers. My friend Jyotishman likes to call it new advances of light in biology. So in the spirit of Diwali, which will be exactly, I think, 2 weeks from today, the Festival of Lights, we start with a Festival of Light in Biology. Okay, so for which we have 4 speakers today. Each of them have 45 minutes, plus there will be 10 minutes or so of discussions. And so that it's reachable, so that people can attend it. We have 2 sessions of it, 2 different modes of joining. One is the Zoom part, and only the registered participants can join by Zoom. The other is public YouTube part, because we thought it will be too sad to restrict these 4 lectures, which are anyway online, to anybody who wants to listen to it. So either you are hearing me in Zoom or you are hearing me in YouTube. So welcome very much. So welcome, and sorry if you wanted to join the actual in-person thing and cannot join. Hopefully next year or something we'll be able to get you in. So a little bit about the organization of the thing. So this is jointly organized by the Fluorescence Society, which few of us set up in order to run the FCS conference every year, and that's going to happen also this year. It's a charitable society registered in India. The second part, the OWLS, Optics Within Life Sciences, is an international organization. It is called International Society for Optics Within Life Sciences. There is a 17th edition of OWLS, which happens every 2 years. So there are 34 years of history in it. I think the first one happened in 1991. It's an international organization which is associated with the United Nations arm, and is based in Germany. It happens so the chairmanship rotates around, chairmanship is voted, and it happens that I am the president of the Optics Within Life Sciences Society as of now, and somebody will be the president later. So together with the Fluorescence Society and OWLS, we are organizing the in-person part of it, but this online part of it is also for between the two of the organizations. It is heavily supported by TIFR, which is the Tata Institute of Fundamental Research, and also by IIT Bombay, where the physical thing is going to happen. And it has been sustained really by a few very dedicated souls. Some of you are organizing this meeting, and some of whom are around. The whole executive committee, and especially the core of the executive committee, which is Professor Jyotishman Dasgupta from TIFR Bombay, Professor Anindo Datta from from IIT Bombay, Professor Shobham Sen from the Jawaharlal Nehru University in Delhi, and I play a little part in it. Okay, so I think it's time for me to introduce some of the stalwarts. We chose 4 of the stalwarts to talk about what lies ahead, in a sense. What's the next thing? What's the next big thing? And in that spirit, we have asked them not to sort of explain things to the tiniest detail, but to give an overview of emerging techniques or the techniques that they have recently worked on. The first of the speakers today is Professor Wahid Sandokdar. He's a Canadian-Iranian physicist and absolutely leading light in the world for the area which has come to be known as nanophotonics. So any paper, you type nanophotonics in Google and try to search, and you'll see Wahid Sandokdar coming. In many ways, it's not just one technique or something. So interaction of light with matter at the nanoscale, that's what he is a pioneer of. And today, we asked him that, of course, he could choose anything that he wanted to work on. And today, he has chosen to work on cryogenic super resolution microscopy, which I haven't heard him talk about at all. So this will be new for us, but I'm sure this is something that we're going to hear a lot of from the literature, from scientific talks in the next 5 years. And that's the idea. Let's learn about things which are just coming online and things that will be exciting in the next few years. So that's all. Thank you so much for gathering. Today is a weekday, I know. So thank you so much for all of you who are listening to us on YouTube and on Zoom. And we'll have 4 lectures today, one starting in a couple of minutes. Then we'll have a break for lunch. Then we have a lecture 2 to 3 and by Professor Gilad Haran of Weizmann. Then 3 to 4, these 2 lectures is one after the other. New advances in fluorescence correlation spectroscopy, Professor Georg Enderlein. And then finally, in the evening, somebody will be speaking from the US, mechanotransduction at single molecule resolution by Professor Taiji Bha. Okay, that's 5 to 6. We'll have a break at 4 to 5. Okay, so again, enjoy. That's really what we organize it for. Enjoy, go deep. Those in the Zoom session can ask questions, and please do. And I hand it over to Professor Wahid Sandokdar, unless Jyotishman, the moderator, has something to say. So Wahid,
No,
please go ahead.
I'm okay. Wahid, please go ahead.
Okay.
Okay, you guys hear me well?
Yes.
Perfect.
Does
Perfect.
it sound good? Okay. All right. Well, thank you very much for the generous and kind introduction, and more importantly, for organizing this event and letting me be part of it. So indeed, I'm going to be talking about a topic that we have been working on for actually almost 15 years, but it's been a smaller type of, let's say, activity in our group. So you might not have heard much about it, and we are quite excited because I think that it will make a difference. So we will see. I will walk you through this method, and it will be maybe somewhere between detailed and not so detailed flavor. So I come from the Max Planck Institute for the Science of Light, and before I go on, let me see if I can write. So I kind of tell you where we are. If this is the map of Germany, Erlangen is a town of about 100,000 people, a big university, but it is particularly known for some major industrial players like Siemens Healthcare and many other branches of Siemens, Adidas, Puma. These companies are all in Erlangen and its immediate vicinity. But probably the most important thing in Erlangen for us is our Max Planck Institute for the Science of Light, which was established in 2009. It's still one of the newer Max Planck Institutes within the Max Planck Society. What's particularly exciting and maybe also relevant to those who are interested in biology and medicine is that we just established a new center as an extension of our institute, and this is what we call the Max Planck Zentrum für Physik und Medizin. So it's a research center to get physicists to address fundamental questions in medicine or in medical research. And this new building was just inaugurated a few weeks ago, and it's right in the middle of the university hospital. So we're quite excited to bring the type of techniques that you're familiar with into medical research. All right. We also have a graduate program. I think this might be a good opportunity to do a little bit of advertisement for it. The topics are very broad, going all the way from quantum optics to biophotonics and medical optics. So if you're interested in any of these things to pursue a master's or a PhD, then feel free to contact us via these different channels. All right, with that little bit of general introduction of the localities, let me start by tuning in to the topic. So we're going to be talking about microscopy, and especially super resolution microscopy in a slightly new way. Of course, you're all familiar with different length scales that might appear in a biology type of context. And over the years, there have been lots of different methods to explore these different length scales. One used to say that light microscopy is limited to the diffraction limit, and now one knows that there is a method called super resolution, and I'll say a little more about that. That takes us down to say tens of nanometers, or more recently to a few nanometers. But and this is the kind of thing that happened in the last, say, 30 years, slowly. That optical scientists were able to take us from the diffraction limited spot of the order of a couple of 100 nanometers per visible light all the way to a few tens of nanometers. And that was a breakthrough that was important enough that deserved the Nobel Prize in Chemistry in 2014. But if you look at this logarithmic scale, then you see that if you want to bridge all the way to the atomic scale, there's still 2 orders of magnitude left to bridge. You will hear a lot about FRET. So FRET is not an imaging method really, but it takes you to a few nanometers. But even from there, you still have another order of magnitude to go. And that's kind of what we have been focusing on. And it's going to be the topic of today's talk, how to go with optics to the angstrom level. All right, so a quick review of the basics. So if you have one molecule or a spot or emitter, you get a spot in your camera. If you have two of them and they're close, then you get a blob and you can't tell the difference between the two. And this is the Abbe formula that tell you how it's related to the wavelength. Now, an important breakthrough came in the early 90s when people showed that you could detect single molecules. Now, detecting single molecules per se doesn't have anything to do with resolution. It's more of a sensitivity issue. But the ability to detect single molecules was crucial in developing many of the super resolution methods, and in particular, what we're going to be talking about. So Eric Betzig and W. Myrna won the Nobel Prize together with Stefan Hell for super resolution microscopy, but I consider these two gentlemen as contributions mostly to the early work that they did in terms of just the detection of single molecules. W. Myrna did that at low temperature and Eric Betzig at room temperature. So the idea of super resolution microscopy mostly is that you do localization. That is, you start with a single molecule, you have an image of a single molecule. Obviously, it looks kind of noisy. But if you were to fit this with a fit function, something in the simplest case, a Gaussian, then you can find the center of that. And the precision with which you find that center is just a question of your signal to noise ratio. So now the question is, how do you do this? How do you take this to resolution? That is, if you have lots of molecules. And the idea is that if you have lots of molecules, not just one, you find a way to only look at one at a time. And what you see here is a little simulation movie that shows how individual molecules are lighting up in a structure that on the left would be just imaged in a standard optical microscope. But if you were to find out exactly where each individual molecule is, eventually you will see that it becomes something like a Christmas tree. Okay, so of course that's an easy concept, but the key question is, how do you manage to see only one molecule at a time? And there are different approaches to that. The first approach, which you might not know about in this community, was actually the approach that was pioneered by W. Myrna and Michel Aury in the early 90s. That's a cryogenic experiment. They had to go to liquid helium temperature to look at individual molecules, and individual molecules now are found by doing spectroscopy. So what you see here is a crystal, an organic crystal, and in this organic crystal, this is a crystal of anthracene, and in this anthracene crystal, you have this type of molecule, dibenzotyrrolene, that are doped. And because each molecule sits at a slightly different site, they have different resonance frequencies. And when you go to very low temperature, the resonances become very narrow. So if you scan your laser, as this little animation is showing, then you will be exciting one molecule at a time. And of course, if you have an imaging capability, then you will see that molecule one at a time. So this was actually a method that was demonstrated in the mid-90s, even in microscopy in that way, but it didn't catch people's attention much because it is done at very low temperatures, and it didn't really talk to standard biological applications. The breakthrough that led to the work in 2014 was the Nobel Prize in 2014 was photo activation. So either using fluorescent proteins or other dyes, if you could have a means to optically activate one molecule at a time, then you could image that and find the center of it and then go on and do that type of resolution that we just talked about. In this talk, I'll be talking about exploiting stochastic photo blinking. This is a property that was also discovered in the 90s when people looked at single molecules, that every fluorophore somehow doesn't always fluoresce, but it goes to a metastable, for example, a triplet state, but there could be other reasons for this. So there was a period in which the molecule is dark. And this stochastic blinking allows us in our experiment to not have all the molecules on at the same time so that we can catch them individually. Now, I mentioned before that it's a matter of signal to noise ratio, so I want to impress you more visually. What you see here is on the left, an image. If you were to have a signal to noise ratio of 5, then you see that it's clearly noisy, but that already would allow you to do localization to within 10 nanometers. But if you were to improve your signal to noise ratio to 30 and 200, then there's no limit to how well you can find the center of that molecule. So in this case, you see that you can do that to within 5 angstrom. Now, the reason the super resolution experiments in the first round did not do this is because there is photo bleaching. And before you get a chance to integrate for long enough to get a very large signal to noise ratio, photo bleaching kills you. So typical room temperature super resolution experiment ends up having a resolution of a few tens of nanometers. So at low temperatures, we showed, but it was already known before that generally, that of course photochemistry is slowed down because you are at very low temperatures and there's no, let's say, thermal activity for different atoms and molecules to meet each other and undergo photochemistry. And this graph shows some of the early measurements that we made to show that, for example, at room temperature, a single molecule typically emits of the order of a few million photons before it bleaches. And if you go to 4 Kelvin, that's extended by several orders of magnitude. This is a logarithmic scale here. And this extension of the number of photons that are emitted by one molecule is the key to the work that I'll be talking about today. So this is basically the same physics demonstrated slightly differently. So we're comparing room temperature and liquid helium, but now we're not talking about the number of photons that were emitted before the molecule died, but in real units of time, how long it takes for a molecule to die. So you see that you typically at room temperature you can work with a single molecule within a few tenths of seconds, and if you go to low temperatures, this becomes hours. So within the shot noise limit, if you have more photons, you get a higher signal to noise ratio, and that allows you to get a better localization. And these are really just measurements that were made. You can see that if you kept looking at the same single molecule, when you collect few photons, your localization precision is not that good. And as you collect more, then your localization precision becomes better, according to this 1/square root of n, which comes from the shot noise limit. Okay, so slide about a little bit of the technical details. So we're talking about cryogenics, so that means that you have to do your experiment in a cryostat. You can actually currently even purchase a cryogenic optical microscope, but these usually go to liquid nitrogen. And there are even devices that you can put under your normal microscope. So we started with such a device. This is the bottom part. That's a cryostat that you can usually purchase. It's a liquid helium cryostat going down to of the order of 4 Kelvin. And this bottom part here, you can just put under your microscope. It has a very thin window so that your microscope objective can be at room temperature under ambient conditions. And through this very thin window, you can look at a sample that is at of the order of 4 Kelvin. We have modified this by extending this chamber, adding this chamber on top, and the inside of that looks more like this. So we have a piezo scanner, and we put the microscope objective into the cryostat because it gives us the freedom to have a higher numerical aperture and get closer and generally have a more stable cryogenic system. These are all the details of how to approach the microscope objective, and again, how to move it around. This is another view of how things look. The bottom part is the commercial part that I told you about. There's a cold finger. We have modified that a little bit. And then there's a microscope that comes in the upper chamber. And this is another view of a little bit of the details of how some of the homemade mechanics combined with commercial piezos allow us to have a coarse and fine adjustment of the microscope objective. Okay. Now, I mentioned before that we have to do use photoblinking. Photoblinking is something that is fairly well studied, but not 100% understood at room temperature. But its properties at low temperature are not as well known or appreciated. And there is temperature dependence to it. What we discovered, which is fortunate, was that by going to about 4 Kelvin, photoblinking actually has just about the right features for us to be able to use it. So what you see here is a very long trace of fluorescence. And if you zoom into that, then of course you see the intermittency of fluorescence. So these are the periods that the fluorophore is on and the periods that the fluorophore is off. If you look at the time distribution, this is a stochastic process, of course, but if you look at the time distribution, then you see that there are on times and then there are off times. And this, of course, is different for slightly different for different molecules and under different conditions. It sometimes depends on the matrix in which they're embedded. And it also depends on the intensity of light. So here you see 3 different measurements at different intensities, changing the intensity by an order of magnitude. You see that this on-off ratio becomes more favorable when you go to higher intensities. And one thing that you want to have here is that you want to have long off times so that the different fluorophores don't interfere with each other. Okay, so the general strategy now is going to be that you take something like a protein and put different fluorophores on it. And now the challenge is to actually find where those individual fluorophores are. And in that way, what I'm going to be talking to you about is we originally called that cryogenic colocalization in 3 dimensions. with an acronym COLD, which seemed quite fitting. But if you look at it more in the context of, let's say, electron microscopy, then it might be good to call that single particle cryogenic light microscopy in analogy to single particle cryogenic electron microscopy, which has become a very powerful and useful method. And with the single particle, I'm emphasizing now that when I talk about super resolution, I'm not interested in resolving details of a whole cell. We want to go to angstrom type of resolution. And at that point, you really care about very small details. So you don't look at the whole cell at the same time. So the idea is to take something like a small or large biomolecule or biomolecular complex, and then study and learn something about its structure and conformations. Now, if you had these three different labels and ran the movie, then each particle would be blinking in your field of view. So this is a typical kind of movie that we record from our cryogenic microscope. Again, you see that each point is blinking. Each point is a diffraction limited point spread function. And at this level, we can't tell anything about the details of those individual fluorophores on one particle. But if you think about that particle and the fact that the particle in general can be oriented randomly in the matrix that it's embedded in, then its projections would look different depending on its orientation. So using that technique, one of the first things that we looked at was to use just two dye molecules and label them to particular sites of this PAS domain of histidine kinase. And the reason we chose this was partly because of the interest of our collaborators, my colleague Christian Kliesinger at the Max Planck Institute in Göttingen, but partly because these two sites were supposed to be really very small. You see that the length scale here is only 1 nanometer. These are already the end results, but I'm going to walk you through how we get to these. So if you have two dye molecules on one particle, then in general, you might have 4 different intensity levels because the two dyes are not 100% identical. They might have different polarizations. They might have different quantum efficiencies. So they don't necessarily give the same amount of signal, although they're supposed to be nominally the same molecule. So they could be both off, that's the lower level. We could have a situation that one of them is on, or then the other one is on. And of course, it could also happen that they're both on. So these 4 levels are there. And now if you were to focus on only one of these middle levels, in those situations, you're only seeing on your camera during the on time, only one of those molecules. And by doing that, you can find different projections. So these are 6 examples of 6 different particles in the sample, and they have different projections. Each spot or blob in each one of these fields of view shows you the position of one of the dyes, and its size tells you the precision with which we could find that dye. You see that the sizes are different because the quality of the image is not always the same. So we assign a localization precision to each measurement. And here's another example in the same work where we took 4 dyes and we put the 4 dyes, attached them to the binding sites of biotin in streptavidin. So there are 4 biotin sites and we labeled them with dyes. And now you see that the projections have 4 points, and these could look completely random, but when you put them all together, you can do a 3D reconstruction. And again, these algorithms are originally developed by electron microscopy people who do tomography. So up to this point, this work was published in 2017. Again, we started it around 2009, 2010, and we published this work in 2017. And we were using, up to this point, really these different intensity levels. And we improved on that some by exploiting polarization. So if you have, for example, two dyes, these two dyes are oriented in a certain way, and contrary to what you might be used to in solution phase, they no longer rotate. So the dipole moment is now fixed. But it could be almost anything, because the way you label them and the way they are embedded, they are randomly oriented. So now if you were to put these two dyes at different places, they will blink as before, but every time, in each blinking time, not only there is a slightly different intensity, but also there's clearly different polarization. And so if we send the emitted light onto a polarizing beam splitter and split that into two orthogonal channels and image them simultaneously, then we'll be able to record each one of these separately. And this gives us a better ability to separate the two signals from each other than before. Before, if you remember, we had intensity traces, and obviously they are noisy within the shot noise, so it's a little more difficult to find out who is who. But here, if you do this, then the polarizations can be separated extremely well. So this is now a trace that in which I'm plotting the polarization and not the intensity. And you see that in this case, we have two polarizations at 75 degrees and 35 degrees, and they're very nicely separated from each other. The intensities of each one, blue and red, will of course overlap, and each one of these would have a center, but it's easier to do the assignment, or it's more reliable to do the assignment by looking at the polarizations in your whole video series. So this is what we then demonstrated in 21, and in that work we used DNA origamis as nanorulers. So here you see a proof of principle where you go from a large distance of 95 nanometers to a small distance of 6 nanometers, and you could show that you can really find each time the distances to a very, very high accuracy and precision. The error uncertainty that we have in localizations are shown here. They're all in the angstrom regime. All right, now that was all more proof of principle. At that point, Hisham Mazal joined our group, and he was particularly interested in exploring this technique applied to different proteins. So this is the one protein which has a trimer type of structure, and we're going to be attaching 3 dyes to these 3 different sites and play the same game as before. So again, we are embedding these as before. Maybe something that I forgot to mention or emphasize, we're embedding these proteins up to now always in PVA. In PVA, so a thin polymer film. And then in the more recent work, we also have a gold layer that is spaced by a silicon dioxide layer to make sure that there is no quenching. The gold layer acts a little bit like a mirror to get more signal, but that's a little bit of a detail. So these different proteins are oriented randomly, and as before, we try to identify them. But now you see that although we have 3 of them, these are 2 examples of 2 different particles. You see that the intensities might be close to each other, but the polarization can be resolved quite nicely. And by doing this, we could find the projections again. And each time you now see a simulated or a fit corresponding to each one of those measured cases, that tells you basically what the orientation must have been for the projections to have turned out the way things turned out. And again, the size of each one of these is telling us the localization precision. And you also notice that sometimes in one image, you have a mixture, and one molecule gives you a better precision than the other. So if we look at all the precisions that we have, it would obviously have a spread. Some of them are extremely good, only a few angstroms, and some of them go all the way to maybe 1 to 2 nanometers. But in the analysis, you can always pre-select and not look at the ones that have slightly worse localization precision. So when we want to really have high quality information, then we throw away many of the data points that turned out not to work out as well. So those are localization, distribution of localization events per dye. And this is now how a distance distribution would look. The distribution, that means that we're now measuring distances between each 2 dyes, and then it would have a distribution as such for the structure that we have. And the dominant side length is of the order of 9 nanometers. But as you project them, of course, you'll get a whole bunch of smaller distances, and that's what's projected here. And of course, you have to account for your precision with which you find these distances. And when you do all of that, then you can actually fit that with the red curve. And then by doing that, we can extract the side length for that structure. So this is the situation. Then afterwards, I mean, you see that the structure, of course, is not from us. This is the structure that is known from the literature, but we have put dyes at these positions, and now we can figure out where they are sitting, and this distance we measure to be 9.9 plus minus 6 angstroms. You can, I showed you before that you can assess the localization precision, but you can also assess the, let's say, the uncertainty that you have in measuring distances. This is something that is quite often done with a Fourier shell correlation method. You basically take all your data and divide it in 2, and then do a correlation between these 2 randomly chosen subpopulations of your sample, and see how well that correlation works, and then compare that to a certain resolution criteria. In this case, it's a so-called half-bit criteria. These methods have all been developed from electron microscopy, and when we started this work, our goal was to develop an optical technique that rivals electron microscopy. On the one hand rivals, on the other hand complements, and so we're trying to make this as compatible with electron microscopy as possible, and also our strategy is such that we can use many of the wisdoms that have been gathered over the years from high-end electron microscopy. Okay, the other structure that we have looked at is a hexamer. So now we have 6 points that we could label, but the technique that I've told you about so far has difficulties going all the way to 6 with high reliability, because by the time you have 6 dyes, their blinkings cannot be separated as nicely from each other. So what we decided to do was to do partial labeling. So you label in a way that on average you only have 3 dyes. Of course, it could happen that you only have 2 dyes. You don't look at those samples. It might be that you ended up having 4. You don't look at those either. But you take the ones that had 3 dyes. And now I'm showing you that in a uniform hexamer, you end up having 3 classes of distances. And these 3 classes of distances should come out in our measurements, and then we can classify them. So the 3 dyes might be sitting in this arrangement, might be sitting in this arrangement, class 2, or it might be sitting in class 3. And in each one of these cases, overall, you would have distances that would be 9, 15, or 18 nanometers. So here are some examples of projections again, as before, if you have 3 dyes. And again, you see that some molecules are localized extremely well, some of them less well, but all that information goes into the statistical analysis at the end. And here are the 3 classes that we found. So we do this, of course, by running an algorithm. That is, you establish a model and then run an algorithm to do the classification. And these are the results that we find. And that allows us, again, to know exactly where all these points were on the hexamer. So far, all of this has been really just a proof of principle to show that this resolution is reachable, that we can do co-localization. And so we've looked at mostly known structures and have done a proof of principle experiment. Sorry, let me get out of this. And this work has been, you can read that in some of the original works, but there's also a nice review now that Hisham put together and was published recently. So you can read all about the details. The kind of a take-home message at this point is that we have reached angstrom level precision in figuring out where each dye molecule is, and also in finding distances between 2 dye molecules. But of course, each dye molecule itself is larger than 1 angstrom. So the way you actually put the label onto your molecule of interest becomes now important. So the size of the linker, the size of the dye molecule, these are some of the things that have to be taken into account if you really want to extract angstrom information regarding the structure of this molecule. But having come this far, we really, in a way, have done all that optics could do, because at some point now the problem is no longer limited by the resolution of optics, but it's more limited by, let's say, the quality of labeling chemistry. Now, we published this work in 2017, originally reported and talked about angstrom resolution. Last year, there was another paper in Nature by the group of Alf Jungmann, also from the Max Planck Society, from a different Max Planck Institute. This is a room temperature work, and very recently, my colleague Stefan Hell, also from the Max Planck Institute in Göttingen, published a work using MinFlux, getting to angstrom precision. In fact, collaborating with our previous collaborators, Christian Kliesinger, looking at the same structure that I showed you before. So angstrom resolution seems to be now reachable by different methods. But the point is that up to this point, everyone has done it using chemical fixation or putting in a polymer. And of course, if you're really worried about angstrom type of detail, then you want to also worry about sample preservation at that scale. Because if in the process of chemical fixation, something has moved by a nanometer or several angstroms, then of course you might have a very good measurement, but you're not getting all the information that you should be getting. Now within the
Sorry, 5 more minutes.
I'll be done in 2. Thank you. So cryogenic preservation is something that was developed again by electron microscopists, and although cryogenics might seem not very biological, it turns out that the best preservation of your biological sample is done if you shock freeze your sample. And there are different methods for doing shock freezing. We have established this technique now, so we've extended our cryostat to allow for shock freezing a sample and transferring that shock frozen sample to the cryostat. So this is the cryostat, this is the transfer system, and you can do shock freezing using a commercial station, and then afterwards we put that in here and transfer that. I'm not telling you the details about that, not just because of time, but also because we're just about to submit a couple of papers using this technique to really look at important transmembrane proteins in their native state using this technique. So stay tuned, and hopefully by next year we will have fresh results. So these are the people who have contributed over the years. So it's really been a small effort in our group, starting with Bo Jing, who did his master's thesis when we were still at ETH Zurich, and Alois Len was a staff scientist at the time, helped Bo. In Erlangen, Siege Weisenburger was really the person who got this whole method to come as far as it has, and Daniel Burning was a postdoc who, together with Franz Wieser, introduced the polarization technique, and now Hisham Mazal is a very, very strong driver of this technique and developing all these things that will be soon hopefully published. There is one flash that I make for those who might be interested in completely different things. So you will have talks on FRET and FCS and all of that within this type of meeting. And you might want to consult this paper that was published a few months ago, where we show that by having a particular arrangement, which we call an optofluidic antenna, you can collect a lot more photons from your fluorophores in solution. So with that, and let's not worry about this, with that I stop and just advertise some postdoctoral positions that we have in our group if people are interested. Thank you.
Thank you so much for a wonderful lecture. We learned a lot. So, so we have about 10 minutes or so for question and answer. Since I may not get a chance later, I will ask one, and then we'll go and see. So people, you put your questions in the chat box, and we'll take them as many as we can, one by one. So the question that I have, and probably is in the minds of many, that it's wonderful that we have come 2 orders of magnitude from the dimensions which won the Nobel Prize, which is fantastic. We're reaching an angstrom level, single angstrom levels. Now, the fluorophores themselves are many angstroms, okay, tegrodamine or something. So when you're talking about localization down to an angstrom level, whose localization are you talking about? It's like the centroid of the pi cloud, or how do you connect to the actual fluorophore?
Right. So that's a good question. What you end up imaging is a dipolar transition in your dye molecule, and of course, in each dye molecule, this could happen at a different corner of the molecule, let's say, and that really has to do with your electron cloud and the combination of what is exactly your LUMO and HOMO, and where the transition really takes place. And so theoretically, you could even have a molecule that has several different optical transitions in different corners of it that would be thinkable. And then you would find each time the center of that dipolar transition.
Thank you. So again, people who, if you have any questions, put them in the chat. But since I don't see one right away, I'll ask a second question. My privilege.
Shudipto,
Yeah.
there is one chat question which came long back from Dev Shankar. So
Okay.
I will
Take
read
it
that
out.
out. Maybe I can just read that out.
Read it out.
Yeah. How does this technique compare with MinFlux, by which you get around 1 nanometer spatial resolution, but also in live samples at room temperature, and therefore ideally can get conformational dynamics? So that's the question, Wahid.
Right. So that's a good question. I have difficulties comparing these because I haven't seen a very quantitative critical analysis of MinFlux performance. But you can think about this maybe yourself. MinFlux needs a certain time to integrate, as any other method does. Now, if you have a live sample and things are moving, the chances are that things are moving by a substantial amount on the level of angstrom or 1 nanometer within your integration time. So most MinFlux experiments, like the one that I showed, they're done on fixed samples and not live samples. There are MinFlux experiments that do tracking, for example, and but they're not reaching angstrom or 1 nanometer. So I think you have to choose your kind of what you want to optimize. If you want to really get to the angstrom level, then you have to fix your sample, because otherwise things are jittering around. And but MinFlux in itself, I mean, any of these methods theoretically are not limited, so they all can get to angstrom resolution.
Okay. Do you see any other questions in the chat box, Jadishman?
Yes, there are two questions, but let's first have Orindam ask a question, and then we can go to, there is one question from YouTube also. So I'll just read them out, but please.
It's wonderful, fantastic, loved it. One thing that I wanted to just comment on is that one issue with low temperature, when you go down to cryogenics, is that your objective is going to have a numerical aperture much smaller than what you'd have room temperature, right? So you're going to lose out a lot of photons in that way. Is there any solution to this? Can we somehow manage to,
I think there is a solution. The solution is requires technical development, and we haven't worried about implementing that. So in many of our other cryogenic experiments that are not about biological imaging, we use a so-called solid immersion lens. So a solid immersion lens allows you to get to a very high numerical aperture, and the problem with the solid immersion lens is that your field of view becomes smaller, and you also might run into aberrations a little more easily. So if you really want to do high-end experiments, you can go in that direction. But as you saw, because the molecules live for a very long time, we're not at the end limited by the number of photons that we collect. You could do the experiment maybe 2 or 3 times faster, collecting the same number of photons. But at the end, we get the
That's why
photons.
you're gaining the. Yeah, but the other question that I had was specifically about the PCNA project that you talked about, where you use the polarization result measurements, and you talked about these three dyes, right? 3 dyes are connected there, right? Now, I was imagine that if you have these 3 dyes, I would have their dipoles oriented at a particular orientation, right? I mean, because of the symmetry of PCNA. Right? I mean, if it's flat kind of thing, I would see certain angle, but at different orientations, I'll get a projection. Would it be possible for you to verify these angles that you can obtain from the polarization data? And have you tried to do so?
You can get access to these angles theoretically. You mean out of plane angles, right?
Well, it can be, but I'm talking about the relative angles between the 3 dyes, right? Because if you have it,
so that
because
we
it's fixed,
definitely
right?
have.
Yeah,
Right.
it's fixed, right?
We definitely have those, but I think the uncertainty is with the way the dye is linked to that position. So they are not necessarily gonna respect the symmetry of the structure because the dye sits still the way it wants to sit.
Oh, so because of the loose. Okay. Thank you.
Okay, there are few questions, but there's one YouTube question from Onkona, who is asking if, since the measurements have polarization sensitivity, what happens if these systems are highly fluxional? Do you still get information which is useful for systems which are
Sorry, if the system is highly what?
Conformationally fluxional. So if there is a lot of structural movements, conformationally flexible systems, you will still get the
Again, I mean, we're these things are all frozen, so you end up having a whole bunch of distributions. That is true, but we don't care about that because we only use the polarization information to distinguish the dyes. We don't use the polarization information for anything else. Afterwards, you have an image and you have localized it, and you localize as usual where the molecule is.
Right. Okay. There are 2 questions, one from Kush Kaushik and Shubhajit Chakraborty. Kush, maybe you can unmute and ask the question. It's too long, so maybe a short version of that, please ask. Kush? Okay, I will just give Kush's a question to you. So he's asking, for the super resolution microscopy, we generally use high magnification and high NA objectives. Pixel numbers are very important too, to a certain extent, but here you are using a low magnification, low NA objective in cryogenic system and still getting very high resolution. Could you explain this idea?
Right. So that is somewhat related to an earlier question. The numerical aperture is not super low. The numerical aperture that we have is of the order of 0.9, but still lower than 1.4 or 1.5 that you would use at a good room temperature experiment. But at the end of the day, you can do super resolution microscopy with an arbitrarily bad numerical aperture. It's just that your let's say your Gaussian becomes broader and broader, and then you need more and more signal to noise ratio to find the center of it. So there is no fundamental reason why you need a very, very high numerical aperture. It's just very helpful to have the higher numerical aperture because then with a fewer number of photons, you get to the signal to noise ratio that allows you to reach a certain uncertainty. And in our case, what we're showing is that our setup is sufficient to get to this angstrom. If you can improve your numerical aperture, then again, you will be doing better.
Okay. There's a question from Shubhajit Chakraborty. He says, thank you.
Jyotishman, this will be the last one because we are coming to the next one.
Okay. Thank you for the great talk. I was just curious about a couple of things. What type of MO can you couple with the cryostat? Does it need to be very high working distance? And the other question is, in the single molecule level, while doing polarized measurements, you throw the other type of polarization emission at one detection channel. At very low fluorescence signal levels, how do you take care of the signal to noise ratio?
The first one was regarding the working distance. The working distance doesn't have to be very high because it used to be that we needed a certain working distance because we had a window in our cryostat. But because we have eliminated the window, in principle, the working distance is not the problem. The only thing is that we're using an air objective, and that automatically puts a certain restriction in the way you choose your objective. So the working distance is not the problem. It doesn't have to be particularly high, and it's not very high in our case. And the second question was regarding
the second
polarization.
Question, yes, it's saying that you're throwing away information if you're just doing some polarization measurements. So if for low fluorescent signals, how do you take care of signal to noise?
Oh, you mean because we're splitting it?
Right.
Well, to some extent, yes, and to some extent, no, because I mean, we have a lot of signals, so we don't really care about this. I showed you that, one molecule just basically doesn't ever bleach. So you can integrate. It is true that, if in many of these questions, if you could do a better thing, then you would do your experiment faster. But these are experiments that I would say that once you get to this level of experiment, and I think that's the same with DNA paint or MinFlux and all of that, you have to see how much effort you put into your experiment to get to this level of information, and also compare that to electron microscopy of how many days of measurement and analysis people do to get to angstrom level. And compared to that, let's say a few hours more or less is just not very decisive.
Sure. Okay. I think we will end here. I'll request Ganesh and Debanjan.
Jyotishman. Yeah, sorry. Very sorry. I realize that it's of course lunch after this, and I really have a burning question.
Yes. Please go ahead. Please go ahead.
All right. So so it's really tough to compete with single particle cryo-electron microscopy because it just gives so much information about the atomic positions and all. However, one thing it doesn't have, and fluorescence will always have, is the contrast. Now that becomes useful when you're not looking at single particles which have been purified and put on a substrate, but can be really important when you are talking about a protein which is, let's say, inside a cell, living cell, which has been frozen, cryofrozen. And has been a long-standing question that in the crowded environment of the cell, is the conformation exactly the same as it is when the protein is in simple solution? And it seems like this would be a great technique to figure out whether, I'm sure for some proteins it is not, whether that is true or not. I mean, which proteins change and which proteins don't. Any idea about whether this can be done and whether you are working on it?
I can only tell you that, yes, we're about to submit a paper, and I can maybe just say that it's on piezo. And we do see things that we think are quite new. Whether it has to do with the fact that we are doing experiment on a native cell membrane compared to previous measurements that were done on model membranes, it's a more difficult question. But definitely, I think this is one of the advantages. That's why we've gone through this whole trouble of developing freezing station and all of that.
Okay, so I'll just end by saying that there are a few other questions from Ganesh and Debanjan. They will ask you via email, Wahid, because it's
I would be very happy to answer them.
Yes. Ganesh and Debanjan, I request you to please email Wahid immediately. And thank you again, Shudipto. Please continue. Please finish.
Yeah, so thanks a lot. A big round of applause for Vahid, Professor Vahid Samdukdar, for giving this, taking out the time early in the morning in the UK for giving this lecture. And we're all so looking forward to having you in November in person so that many of those questions can happen over a cup of tea or cup of coffee in person in
Absolutely.
Bombay. Looking forward.
Look
I'm
forward
looking
to
forward
meeting
to
you.
that. Thank you very much.
Okay.
See
Thank
you all
you.
then.
Bye.
Bye-bye.
Audience, we gather again at 2 o'clock. So you have to get in around 10 minutes to 2 so that we have time for Professor Giladharan's talk on single molecule FRET for studying fast protein dynamics. Okay, thank you. As this session ends, but I think Jyotishman,
I will continue
they
the
stay
Zoom.
long day.
I will continue the Zoom. I'm not switching it on off, so people can join at any time. The only thing is, I want to thank Wahid for a wonderful talk, and we'll keep this in YouTube, Wahid, and we'll share it back with you. Okay, thank you.
Okay, great. Yeah, thank you very much for doing this.
Bye.
Thank you.
Just once more request to the audience, in case you leave and join again, please join with your own name as well as the institute name so that we recognize that you are one of us and not somebody from outer space trying to mess up things for us. Your name as well as institute name, not just your name, not just institute name. All right. So Jyotishman Dasgupta, TIFR. Jyotishman Dasgupta, TIFR Mumbai. That is what you write. So please, yeah.
Anindo, the link that you shared,
yeah,
that is for only our students, right?
yes. Please don't share the link with anybody because we want to make sure that we do not have someone who
I know.
might
The only for outsider is the YouTube link, right?
Yes.
Yeah.
But still, we have more than 100 people here. It is great. Okay, I think we have earned our lunch.
One problem I faced, I could not put the question to the chat.
Yeah, I also didn't see any question in the chat. So yeah.
So
in the
it
chat
says,
box, I think the permission is to be given.
yeah, so due to large number of
Let me
participants
see.
in this meeting, system messages for those who have joined on left have been disabled.
Jyotishman, you can enable that maybe. I think only you can do that. I don't think I can.
Yeah, I will try to do that. Yeah.
Okay. All right. I think we have earned our lunch. Let's enjoy it. And we'll
be
start
back
again.
at 10 minutes to 2.
Yes, to 10 minutes to 2. See you. Bye-bye.
Bye.
Hi, Jörg.
Hello.
Hi, Jyotishman. I don't know if you remember me. You came long back, almost 8 years back
Yeah,
in
yeah,
TIFR, and it was a fantastic trip for you because you gave a evening lecture, if you
yeah,
remember, in the lecture theater. So
yeah.
yeah.
Nice to
So
see
good
you.
to see you after a long time, and I just wanted you to check once if your slides are working.
Yeah, yeah,
Yeah. Hello, George. Good to see you after not so long a time. We met last
yeah.
week.
Exactly. So let me share my screen and we can.
Yes.
So I have to start my presentation. Okay, good. And then so here then I think this is this one. Can you see something?
Yes.
Super.
Yes. Mhm.
Yeah, so that would be then, yeah, just one slide. Yeah. Okay,
Okay.
good, perfect.
So maybe is that the first slide or
No, the first slide is here.
okay, cool. Yeah, we can see it.
Yeah,
Cool. Okay, so it
okay,
works. Yeah, thank
good.
you. Thank you, Jörg. Yeah.
Yeah,
So maybe in 40 minutes, we will be back in 40
yeah,
minutes. Yeah.
sure.
Oh, actually 40 is a little bit too long. Maybe around 35 minutes. Is that okay, Anindada? 35 minutes.
Yeah, I mean, my talk is anyway in 2 hours only, no?
Your talk is in 2 hours.
Yeah,
Yes.
yeah. Mhm.
Yeah. So you can, yeah,
I will
you're
simply
fine.
listen.
You're fine.
Yeah. Okay, then see you.
Yeah. See you. Anindada.
Oh, I'm muted. I didn't realize that. Okay. All right. All
Yes.
right.
Yes. Anindada. So everything is okay. I think we are
Yeah.
waiting for Gilad. So I will have some lunch and come back in like 15 minutes.
It's on. So if Gilad joins, I'll be able to see him.
Sure. Okay.
Okay.
You had lunch?
Yeah. My lunch is online.
Do we have Professor Haran yet?
I don't think so.
WhatsApp him and see.
Hello. Hello. Hello.
Your
What?
name is not Gilad Haran. I'm looking for him.
Yes. And also, you're not signed in with your institute name, so I'm going to remove you.
Then the thing will collapse. Rename. Your name is not Giladharan. Baba, thank you. My name is not Giladharan. He's in tremendously tough time. We can't be in his shoes.
Or his bunker.
He's public, right? We are public. All the so
Okay. What?
sent him a WhatsApp, so hopefully he will join soon.
Okay. So people are coming back. Number is increasing.
So how many registered participants do we have?
Around 280.
Okay.
So almost 3/4 or more have joined. I saw the number crossing 150.
Yes.
150.
Very good.
Actually, one thing has happened. Our Indian speakers of OWLs are not really in our registered participants list yet. Most of
I
them.
see. That's a bit of a problem.
So we have missed out on them. But of course, I mean, it was advertised widely. So some of them might have joined.
No, many speakers are joining via YouTube. So that's good.
Yes. Dushman, please let people in.
Okay. Again, the same issues exist.
What? You cannot help it. But for tomorrow, we have to send again.
Yes.
Ask Komal to join with her this thing.
Yeah.
With TIFR. Only 3 people are right now there who are in the waiting room without any affiliation. I think it's better than the morning.
Send messages to them.
Yeah, we are doing that from time to time.
Yes, we are doing that quite a bit.
We have only 3 left, and at least one of them is joined with the institute name. Only 2 left, out of whom one has joined with the institute name as well. Okay, nice. Where's Gilad?
He hasn't seen my message. I might call him and see.
Jorg has checked his talk, Shudiptada.
Jorg has?
Checked,
Sorry?
already checked his talk.
Oh, I see. Okay, fantastic.
Yeah. He's in the Zoom.
Yeah, I'm here.
Here he is.
Hi, Jorg.
Oh. Yeah. Okay. $84. So out of 84, 3 are in the waiting room, 81 are in the meeting.
He hasn't seen my message. He didn't pick up my call. So I'm not so sure what's going on. is it possible that if Georg is ready, then we can, suppose he doesn't join in another 3-4 minutes, would it be okay, Georg, for you to go first?
Yeah.
You can wait for 5 more minutes. Yeah.
Okay.
Gilad
Okay.
is here. Gilad is
Oh,
here.
Gilad is here. Okay, good.
Okay, good.
Great, great.
All right. That solves everything. Okay, so welcome back, everybody, to the afternoon session. Hope you had a good lunch. Definitely, we had a lot of scientific things to eat, to our heart's content. In the first session, we heard about angstrom resolution imaging. And now we're going to hear about dynamics. So those are all frozen stuff that Professor Wahid Sandagh talked about. And here we are going to talk about dynamics. And who we have is Professor Gilad Haran from the Weizmann Institute of Science. So Professor Haran has been at the Weizmann Institute for some time, so has risen through the ranks, became a professor, I think about 15 years ago or so, and then became its dean. And he has multiple awards to his name. The one that he received last year, I remember, is the Nakanishi Award, and there was a Weizmann Award, so I'm sure he has lots of awards. But what is most important is that he has done fantastic work, pioneering work in the area of single molecule FRET microscopy and spectroscopy, looking at protein dynamics. And the second most important thing, which is, we are thanking, all of us will thank Gilad, that he has really been a good supporter, great supporter of this FCS series of meetings. He hardly ever misses a chance to come here in person, and we're hoping that he will be able to make it if the Middle East situation doesn't deteriorate next month. So here is your chance to hear Professor Gilad Haran, and he will talk about single molecule FRET for studying fast protein dynamics. Gilad, you want to put up your slides?
Yes.
Hi there.
Okay, can you see it?
Yep,
Yes.
looks good.
Okay, good. Good afternoon, everybody. Wow, it's really impressive that you have so many people in the audience. I guess close to 100. That's quite exciting, and I'm very glad to have the opportunity to talk with all of you. Since I'm going to be also, as Sudipta mentioned, I'm going to be in the workshop next month, and then I will give a talk also in the OWLS meeting. So I decided to give a talk that will be quite didactic, but not get into too many details, I hope. I will try to talk like Sudipta suggested for 45 minutes, maybe even less. And then I will leave you guys time for questions, but you can also ask questions in the middle of the talk. I guess that there must be a way to do that somehow.
Gilad, if I can request, probably that's not a good idea. There are just too many people through Zoom, and it's best that they wait for till the end. Okay, and there are also a large crowd in YouTube.
So Dipta, you are the boss, and so I see that now there are 108 people, so that's okay. So we will do it in the end, as you suggest.
I'll give you a 5-minute warning at 40 minutes.
Okay, okay.
All right, go ahead.
So what I want to talk today about is really about protein dynamics, and we are very interested in the fact that when we look at proteins, often we see very fast dynamics, and very fast meaning that it's on the sub-millisecond or microsecond time scale. And it turns out that that these fast dynamics are nevertheless important for the activity of the proteins, and I'll show you some examples for that. But the kind of the crux of the talk is that we have found that we can actually look at these dynamics and trace them very nicely using single molecule FRET spectroscopy. So I'm going to start with FRET and single molecule FRET spectroscopy and build it from there to look at the dynamics of a couple of examples of proteins, and then we will touch on another method, which is also useful for FRET, but it's not the method that we use. And you will see a little bit of the variety of methods that can be used in order to look at fast dynamics. Okay, so I want to remind you in the beginning about, a little bit about FRET. I assume that most of you in this audience know what FRET is, but since this is a didactic talk, let me start with the idea of FRET. So imagine that you have on a single protein or a single element, you have two dyes, a green dye and a red dye. And these two dyes turn out to speak to each other. And speaking to each other means that when we excite the donor, we actually get, this is the donor, the green one, we actually get some of the energy transferred to the acceptor and being emitted for the acceptor. So we have donor and acceptor, and we look at the emission from each of them. And you can see that when they are very close, there is a lot of discussion between them. Energy is transferred. And therefore, most of the light that is emitted is red light. And so we can see that when we use two detectors to look at the light. On the other hand, if the distance becomes larger, then the situation changes. The green dye, the donor, emits quite a bit of the light, and the red dye emits less of the light. And so again, by looking at the two channels, we can actually register exactly how much light was emitted in each of them, and then get a feeling about the distance. As I will show you in a minute, we can actually calculate the distance quite nicely and accurately. Here's an example when the distance becomes very large. This spring got very extended. So most of the light is now from the green channel because there is barely any talk between the two dyes. So this is kind of a very rough and general introduction to what fluorescence resonance energy transfer is. As you might know, FRET was discovered in 1948 by the German scientist Theodor Foerster, and he also showed how to understand analytically FRET. For that, he said that basically each of the dyes is represented by a dipole moment because you have transition dipoles due to the excitation of the dyes. And so the two dipoles interact with each other. And you can write down, you can use quantum mechanics. Those of you who are familiar with quantum mechanics and the basic tenets of quantum mechanics, you can use perturbation theory in order to discuss the interaction between the two transition dipoles. And you get an expression based on what is called the Fermi golden rule in order to describe the rate of energy transfer between the donor and the acceptor. Then you can take this expression, since we don't have time to really go through the whole expression, you can take the expression and you can express it in different ways. For example, this is a very familiar way to express the energy transfer efficiency rate, energy transfer rate. And this is in terms of the rate of emission of the donor in terms of the distance between the donor and the acceptor and to the power 6. And I just want to remind you that the distance comes into the problem because we are talking about interaction between two dipoles. So if you open your favorite electrostatics or electrodynamics book like Jackson, you will find that the two dipoles that interact, they have their interaction is dependent on the third power of distance. And since we measure intensity here, we look at the square of that. So it's the 6th power of the distance. And then there is this factor here, which we call the first order distance, or R0. And this takes into account actually all the different constants in the problem. And here's how it looks like on the bottom here. And R0 is can be written as an integral over the emission spectrum of the donor, the absorption spectrum of the acceptor, times the fourth power of the wavelengths at which each point here in the integral is taken. So this is easy to do once you can measure the emission and absorption spectra of the donor and the acceptor respectively. And then you get this integral, and then there is another factor which is called kappa square. And kappa square is just an orientational factor which takes into account the fact that, remember, I told you that we have two dipoles interacting with each other, and since the dipoles are like arrows, then they have some orientation between them. We will talk about this in a minute. So we can actually take this rate equation and turn it into an efficiency equation. So this little expression here answers the question, what is the efficiency of the energy transfer from the donor to the acceptor? And of course, efficiency goes from 0 to 1. And so the question is, what part of the energy that we put into the donor goes into the acceptor? And you can see that it is related again to the distance between the donor and the acceptor, and to this first-order distance, which includes all the different factors. Now, the efficiency has the nice property that you can plot it as a function of the distance, and it looks like a sigmoidal curve. You can see it here. As I said, it starts from 1 at very small distances. It goes to 0 at very large distances. And if you put here R equals R naught, you get half. So R naught is really the distance at efficiency of 0.5 right here. So here's the question of the interaction, the orientation of the two dipoles. So kappa square depends on several angles here. I don't want to get into detail into these angles. You can find this in the literature if you want. But if you have two arrows, then they have actually they can be defined by 3 angles, and you have to take into account these angles when you calculate the orientational factor. In fact, in most cases, we assume that the donor and acceptor are freely rotating much faster than the time of the actual interaction between them. And in that case, one can show that for kappa square, one can take the factor 2/3. So this is a very, very common approximation. If you have seen this approximation, you can know now that this is because of the assumption of a very dynamic donor and acceptor. There are ways to figure out whether that's correct, whether that's the case, because remember that we put the donor and the acceptor on, we put it a few slides ago on a spring, but it's really on a macromolecule like a protein or maybe DNA. And so the interaction of the two dyes with the macromolecule might hinder the free motion, but we can check that in the lab. I'm not getting into that. This is something that if you want, you can check how to actually ascertain that the motion of the dyes is fast enough in order to actually allow this approximation of kappa square equal to 2/3. And there's a reference here for one paper where this is explained. It's pretty old, so this has been actually looked at in the literature already many years ago. One more factor that I want to discuss regarding FRET is this interaction between the donor and the acceptor spectra. So here you see 2 typical dyes that we use for single molecule experiments. For the donor, Alexa 488, for the acceptor, Alexa 594. And the first distance turns out to be around 50-something angstrom. So this means that this is really the distance around which we want to probe the interaction. So we want to put it on a protein in such a way that when the FRET efficiency is half, this will be the distance more or less. This is something that we will see in the experiments that I will show you in a minute. But these are just to show you the absorption and emission spectra of the donor, absorption and emission spectra of the acceptor, and the overlap is between the donor emission and the acceptor absorption. and you have to calculate it according to the integral that I showed you before. Okay, so that's FRET. This is really, if you wish, FRET at a very fast time, because I don't have a lot of time to explain it more. Perhaps we will get into it in more detail in the workshop, or not. You can ask me later and I will try to explain. Now let's go to the single molecule FRET experiments that we do. So they are based on, obviously, on the same theory, the same ideas that I just mentioned, but we are going to do it at the level of an individual molecule. And there are many reasons to do it on the level of a single molecule. One of the most important reasons is that we cannot synchronize protein molecules. Each of them does its own thing at a certain rate. And if we cannot synchronize them, it means that if we look at many of them, we get an average. If we want to get some information, we need to look at the molecules one by one. The typical experiment that we do in the lab, I think probably in one of the lectures later, you will hear about different experiments, but the typical experiment that we do in the lab is we let the labeled protein molecule swim in solution, and we let it pass through a focused laser beam. So we choose the concentration of the molecules to be very, very low, so that the probability that more than one protein molecule will be inside the beam is very small. So this means that typically when we do get a signal, we get a signal from a single molecule. And we know that for a large certainty that it's a single molecule. Now, when the protein, what happens when the protein really diffuses into the beam? It gets excited by the laser light, by the focused laser light. So we have the donor, here it's in blue, and the acceptor in red. And so the donor gets excited, and the donor transfers energy to the acceptor, like we discussed before. And we get also emission from the acceptor. So we get emission in green by the donor, emission in red by the acceptor. And so we get what looks like a burst of energy, and indeed we call this a burst. And each time a molecule goes through the beam, we get such a burst. So this is how the experiment will look like. So here we have a stretch of data of about 25 seconds. In real experiments, we take data for many, many minutes, sometimes hours. And you see that from time to time, every few seconds, you get a burst of photons. And the burst is both in the donor channel in green and the acceptor channel in red. Now, we can actually take each of these bursts, and we will not talk on how to isolate bursts here. There are techniques to do that. We can take a burst and we can ask the question, how many acceptor photons, how many donor photons? And we can use this equation here to calculate the FRET efficiency. So the FRET efficiency equals the acceptor photons divided by acceptor plus donor. It shouldn't be B, it should be D. ID is the donor photon. So we have here in the denominator the total, IA divided by the total is the FRET efficiency. How does it look? We can do that for many, many molecules. Typically in an experiment, we have 5,000 or 10,000 bursts, which means 5,000 or 10,000 molecules pass through the beam. And we can take all these values of FRET efficiency and we can plot a histogram. And this is one form of a histogram of the FRET efficiency, the probability to get different FRET efficiencies. So what you see here, and that's quite interesting, is that the histogram is very broad. How do we know that it's broad? This is what we are going to see on the next slide. Before that, you can see that not only is it broad, but it also has shape. So in principle, you would expect perhaps to see some kind of a single peak in the histogram. You see more than a single peak, which suggests that there are several states, several different forms of the molecule in the beam. And this is really what we want to study. What are the different states of the molecule, and what are the rates by which the molecule jumps between these different states? We will talk about that in a minute. Now, how do we know that this histogram is broad? So we need to analyze the properties of the bursts that we get in the experiment. And what we need to think about is the noise that we expect to get on the data. So remember that we discussed, oh, somehow it doesn't go back. Strange. Anyway. Oh, there we go. Sorry about that. Okay, let's forget about this because it doesn't work very well. So the noise on each on the intensity coming from a single molecule can be described by Poisson noise. You might have heard this term before. In a Poisson distribution, the variance of the distribution equals the average. So sigma square equals I. So this is the typical noise that we get on individual photons that are emitted by a molecule. And one can use this Poisson noise in order to derive the noise in a Fred distribution. You remember that the Fred distribution is IA divided by the total, and we can actually calculate what the noise is. So it turns out that this is the expression for the noise. And this is the green distribution here. So you see that it's pretty narrow for a certain number of photons, n. And it's much narrower than the actual orange distribution that I plot here. And this suggests that the orange distribution contains more than one state. Because if you had one state, you would expect just the Poisson noise-based distribution. So we have several states here. And the question is, how do we actually analyze this? How do we get these states? And how do we learn something about the situation of the molecule when it is in each of these states. So it turns out that the best way to do that is not to look at the Fred histogram itself, because the Fred histogram, as you just saw, is pretty broad. It's pretty unstructured, and it's difficult to get information about different states from it. Rather than that, what we can do, we can take each of the bursts and we can spread it over time, and we can look at the arrival times of the photons in the burst. This is a typical burst. So what you see here is in green are the donor photons, in red are the acceptor photons. And what you see is that it turns out that initially, in this particular example, initially there are very many red photons. Then there is a change where there are more green photons. Then there is a change again to more red photons, green, red, and so on. So there seems to be a change between two different states in this burst. One state with a lot of red photons, which means that the donor and the acceptor are close to each other, and one state with a lot of green photons, which means that the donor and the acceptor are far from each other. Now, if that's the situation, can we just look at the burst and we can say, okay, when the situation is that the donor and acceptor are close, when is the situation that the donor and acceptor are far away from each other? And the answer is that that's very difficult to do, because you see, I plotted here this orange line to actually show you where there is a transition between the two states. A state with, let's say, close distance and high Fred efficiency, and a state with large distance and low Fred efficiency. But in reality, were I to remove this orange line, it would be difficult to say exactly where this happened. So we need to resort to some statistical methods that will allow us to analyze the data and tell us where this happens. And in fact, what we do, I'll show you quite briefly how the statistical methods work. But what we do is we do analysis not on a single burst like this, but on a large ensemble of bursts, something like 5,000 to 10,000 bursts. And by doing that, we can get a very good description of the statistics. assuming that we know, or we can figure out in advance what kind of model we have to use. So for example, the way I talked about this burst all the time was that there are two states. So this is a two-state model. This is pretty famous in analysis of the kinetics of protein dynamics. For example, in protein folding, we talk about folded and unfolded. These are two states. In a case of a protein, like I'll show you, where the protein is either open or closed, again, these are two states. So if we assume in advance that we have two states, we can actually use some statistics in order to get information about these states from the data. This is an example that shows you how the data will actually look like. So I showed you this kind of data, but if I do some binning in order to see what is the number of photons at each moment of time, here I think the binning was done in 50 microseconds per bin. You can see that it's amazingly noisy. So just looking at the data, donor and acceptor, or the FRET efficiency calculated from them, it's very difficult to actually say something very accurate about where there is a change between the two states. But after we do the analysis, as I'll show you in a minute, what we get is this blue curve, which tells you more accurately when there was a change between the states of the data. So that's what we want, because we really want to know accurately what happened in the data, and we want to be able to actually get information about the rates of transition between the two states and so on and so forth. So how can we do that? So we use what we call what's called a maximum likelihood estimation. There is a series of methods that use this type of analysis to look at pairs of photons in single molecule FRET. Initially, this was introduced by Attila Szabo and Irina Gopic from the NIH in a series of very nice papers, theoretical papers. And we and others have used similar methods in order to get the rates. I don't want to get into the full machinery of these maximum likelihood methods. Sometimes they are called hidden Markov models methods, because for the dynamics, we use a Markov model. We assume that there are states. This means that there is these states are memoryless and simple, and that's why it's called the Markov model. On the other hand, it's a hidden Markov model, because when we look at the data, we see the color of the photons, but the color doesn't give us information, a one-on-one information on which state we are in, because a red color can be sometimes from the donor, and a green color can be sometimes from the acceptor. So this is why this is hidden. We need to take this situation into account in the analysis. So let me just give you a very brief feeling of how to do this analysis. More information in this paper at the bottom here. So here's a string of photons coming into the two detectors. Some of them are red, some of them are green. So what we are actually doing is we ask ourselves the question, what is the likelihood, what is the probability that we are going to get this stream of photons, assuming that we have a certain model. And the model is, like I said, let's say in this case, two-state model, either perhaps open or closed. And the question is, if the molecule is in one of these two states, open or closed, what is the probability? And there are certain rates for transitions between the two states. What will we get in terms of photons? So is it possible that we will get this? Yes. But the question goes beyond that. What would be the typical rates that will allow us to describe this kind of data? So we need to write an analytical expression for the likelihood, the probability, and then we need to maximize it in order to fit it best to the data. So let's see what I mean by the likelihood of getting this kind of stream of photons. So, yes, here are the states 1 and 2. So there is a fast exchange between the two states. Let's see how we write the likelihood for that. So let's assume for a moment, and I'll tell you how to relax that in a second. Let's assume that we know also which state the molecule is in for each of the photons. So you see that here the molecule is in state 1, and in this state it emits sometimes a green photon, sometimes a red photon. And then it goes to state 2, then it goes to state 1 again, and so on and so forth. So we want to analyze that. So we write the likelihood function. Here's a general description how to write it. We can get into more details if you ask me a question about that. So we start with the first photon. We ask what is the probability that we get a photon at time t1 in state 1. And then at a certain time later, time t2, there is a transition from photon from a red photon to a green photon, and we are still in state 1. But okay, sorry. Before we transfer to the next photon, we first emitted this photon, so we emitted a red photon in this case. So this is the probability to emit a red photon. Then we take the probability to transfer at time t2 to transfer. In this case, we didn't really transfer, but this is a virtual transfer from state 1 to state 2. So we have a certain probability that we can use to write that. And then there is a probability now to emit a green photon. So it's B of G. And we can go like that again and again. We then ask what is the probability that at time t3 we got we got transferred again from state 1 to state 1 because we didn't change the state, and we got a green photon. At time t4 we emitted a red photons, and we are now in state 2, and so on and so forth. So like that, we build a whole string of probabilities for all the states, and we get what is called the likelihood function. It can include, if you wish, I don't know, 5,000 photons, 100,000 photons, as many as come out from one burst. And typically in a burst, there are just 50 to 100 photons, but there is no limit for the likelihood function. So now what we assumed in this function, as you saw it here, is that we know which state every photon comes from. Since in reality we don't know that, we have to assume that we have different possibilities. So we can write all the different possibilities for an arrangement of states: 1, 1, 1, 2, 2, 1, 2, 1, 2, 1, 1, 2, 2, 2, 1, and so on and so forth. And we need to take an integral over all these different possibilities. That integral is the full likelihood function, and we, like is implied in the title here, we maximize the likelihood function to get the actual rates. So the outcome of that is very important because we get several different factors for which we get numbers. So first, as I said, the rates, the rate from going from 1 to 2 and the rate from going from 2 to 1. And second is the Fret efficiency of each state. And the third one is the populations of the two states. So we know everything about the states, right? I'm assuming that we have this model, the two-state model. We know from this analysis everything about the model, how fast do the states exchange, how many, what fraction we have in each of the states, and what is the Fret efficiency of each state. So that's what we get from the analysis. Now with this, we can actually think a little bit about the dynamics of these molecules and what does it mean that we have two states. So typically we're going to talk about a molecule that is either in an active state or in an inactive state. And like I said before, it's either open or closed. When it's closed, it's active. When it's open, it's inactive. and the molecule frequently goes between active and inactive, between open and closed. And then when it's in the active state, there is an option for the chemical step to occur. So imagine that this molecule is an enzyme. It has to do some chemical reaction on the substrate, and that can occur only from the active state. It turns out, and I don't want to get into the details of that, that one can write, you see that there are 3 rate constants. The rate constants are describe the opening and closing of the molecule, and then there is the chemical rate constant. And so one can actually write the total rate of such a reaction in this form, where you have a product of the rate of going to the active state times the chemical rate divided by the total, by the sum of all the rates. And this is quite interesting. One can go one step further, and one can say, okay, what happens? And you will see that this is the example that I'm going to show you from the lab. What happens when we have the 2 rates of opening and closing are much, much faster than the chemical rate? So if that's the case, we can drop the chemical rate from the denominator, and we are going to get that the total rate is just the numerator here divided by KIA plus KAI, but KIA divided by the sum is just the fraction of the active state. So we have here an equilibrium factor, PA, times KChem. So what it says, if you did not understand what I'm saying so far, you just want to understand what it means, is it means that if the opening and closing rates are very fast, compared to the chemical rate, then it doesn't matter anymore what the actual rates are. Is the Ki larger than Kai or not? It doesn't matter. What matters is the equilibrium probability to find the system in active or inactive states. So we basically convert the problem from a dynamic problem into an equilibrium problem, considering the fact that the dynamics, the internal dynamics, conformational dynamics are much faster than the chemical rates. So this is how it looks. You can see it here in this calculation, which comes from this review that we've written earlier this year. So you can see that if we increase KIA gradually and KAI, we take a constant ratio between them. So we increase them, and then we get to a situation where they are much, much bigger than the chemical rate, which is 50 per second. So here you see when they are like 100 or a few hundreds and even 1,000, they are much bigger than the chemical rate. And what happens in that case is that you see that the curve becomes completely flat because the actual rates do not matter anymore. What only matters is this factor here, P of A, which gives us the fraction of the active state. So we get into this equation. So the bottom line is that if the conformational dynamics of the protein are very fast, what we care about is only the equilibrium of the 2 states of the protein, open and closed. So let's see how it works in reality. And I think I'll just show you one example. I have a few examples, but I'll show you one and I'll stop here. So this is an example of a small protein, adenylate kinase. This is the structure of the protein. And you can see that in this structure, I wrote here open conformation because indeed it looks like it's open. And there is a possibility for this protein to actually get into this structure, the closed structure, where these 2 domains, this is called the lead domain, and this is called the NMP bind domain. These 2 domains actually close on the active site, which is somewhere here, and encompass the active site, and if there are substrates in the active site, encompass also the substrates. So these are the 2 conformations. How do we know that? We know that from crystal structures of the protein. And we know a lot about the biochemistry of this protein. We know that it binds two substrates, one substrate on this side, ATP, one substrate on that side, AMP. And we know that there is a reaction between ATP and AMP to form two ADP molecules, or vice versa. The protein is can operate in both directions. And so you see from this description that the reaction occurs only when the protein is closed, because when the protein is open, the two substrates are too far from each other. So this is a talk about single molecule FRET. We can put two fluorescent dyes on the protein at these two yellow beads, and we can look at FRET efficiency and try to understand how the protein opens and closes. So here's the protein now. Instead of beads, we have the fluorescent dyes. And just to mention the work of two very talented people in the group, Chaim and David, who did this work. Before we look at the FRET data, the domain closure data, we want to first understand what is the chemical rate of the protein. And so it turns out that you can do a biochemical experiment and measure a chemical rate of about 400 per second. It's much, much faster. Sorry, it's okay. Let's leave that at that point. It's 400 seconds. Let's see what it is compared to the rates of opening and closing of the domains. So we take the protein, like I said before, we let it diffuse in solution with the two dyes, and when it goes into the laser beam, it gets excited. And when it gets excited, it emits. There is a burst. We analyze all these bursts. Remember, we calculate the FRET efficiency per burst, and we get a histogram of FRET efficiencies. So this is the histogram that we can get when there is no substrate in solution. So you see that the histogram peaks more or less at the low FRET efficiency. There is also a bridge into high FRET efficiencies, and this is not some kind of a noisy bridge. It's real. It's really in the data. We will talk about it in a minute. And then we add a lot of ATP. We see that there is a very dramatic change in the histogram shape into a much higher FRET efficiency value. It peaks at around 0.7, close to 0.7. And you see that it's also quite broad. What happens if we don't have a lot of ATP, but just an intermediate value of ATP concentration? This is what we get, the gray histogram, which is even broader. It encompasses both regions of the FRET efficiency, and it suggests that the protein really goes between open and closed conformation in this case. Now, I'm suggesting, based on looking at this data, and I'll tell you how I know, that this indicates that there is a very fast motion between the two states. It's not only that the protein is sometimes open, sometimes closed, it also undergoes a very fast motion between the states. How do I know that? Because there is a phenomenon known in spectroscopy called motional narrowing. This is what happens when you look at a certain signal of a molecule that exchanges between two states, and the rate of exchange becomes larger and larger. So you start with the dark.
You'll have 5 minutes.
Yes, you start with the dark histogram, and the rate of exchange between the two states of the molecule is slow. So clearly, when you do the experiment, you see clearly the two states of the molecule with FRET efficiency, low FRET efficiency, high FRET efficiency. And now we start increasing the rate of exchange from 10 to 1,000, from 3,000, 7,000, and so on. And you see that when we increase the rate, we start to build a bridge between the two states in the middle, because sometimes the molecule changes between the two states while it is in the beam. So we measure an average of the two states, and this average becomes more and more abundant in the data. And so when the rate of exchange between the two states is very large, we get something, instead of two separate peaks, we get one peak in the middle. And initially, this peak is very broad, just like the gray peak. When the rate is even higher, the peak becomes sharp and it's in the middle, but we seem to be in the range of the orange peak here, where we have a broad distribution. So we can use this and we can see what happens in reality. And to do that, we do the analysis, the maximum likelihood analysis that I mentioned before. And when we do the maximum likelihood analysis, we get two rates, because we assume that this is a two-state model. We get two rates: the rate of opening in green, rate of closing in red. And we can do that as a function of substrate concentration. Instead of looking at the histograms only, where the histogram is very broad and featureless, we get actually two separate rates. And so this is what I plot here, the rates of the two processes as a function of substrate concentration. And what we find surprisingly is when the substrate concentration is very high, the rates become very large. So eventually we are at a situation where the rate of closing, the closing of the two domains, takes not more than perhaps 15 microseconds. So that's the inverse of this rate. And the rate of opening is a little bit slower. It takes 25 microseconds. So both rates are in the microsecond timescale, very fast. Now you want to remember that the chemical rate of this enzyme is 400 per second. So this means that the opening and closing of the domains is much faster than the catalytic rate. We are in that limit that I mentioned in that previous slide, where we need to take into account just the average or the equilibrium of the two states, because the opening and closing, because the two rates are very, very fast. So why is it important? Let me show you what we think happens. We have indications for that. So when we have the two substrates, donor and acceptor, in order to have a reaction between them, they have to be exactly collinear with each other. But what happens if when they bind to the enzyme, they bind not collinearly, but they bind at some angle? So there will be no reaction in this state. However, if the protein now opens, they have a chance to reorient. And if the protein opens multiple times, they reorient again and again until they get to the right conformation for a reaction, and the reaction occurs. So the opening and closing reaction actually allows the enzyme to be enough time in the open conformation so that the substrates can reorient and get to the right conformation. Now you could say, okay, let's just open the enzyme and then the substrates will be in the right conformation. But if it's open, there is no reaction. So how do we know that reaction? How do we ascertain that reaction will occur? We open and close again and again so that there is enough time in the open state and also enough time in the closed state. So that's what happens here. The microsecond dynamics allow the protein to be in the open state for enough time for the reorientation to occur. Okay, so I don't have time for additional information or additional examples for this. Let me just go directly to the conclusion from here. What I try to show you is that single molecule FRET is very well suited to study conformational dynamics. I hope that I will show you more examples, but I did not. We have an opportunity with this method to actually look at very fast rates. The rates can be as fast as the emission of photons by the molecule, so they can be on the microsecond timescale. And we have in our hands methods that allow us to analyze the time result data in order to get the full information about the models that describe the dynamics, the states in the system, the FRET rates of exchange between the states, and the efficiencies of each FRET efficiencies of each state. And we can do that using the information that we measure. And finally, these studies allow us to understand something important about the relevance of conformational dynamics to the actual chemical step of a protein. I would like to thank people in my group who were involved in this and other projects. And thank you for listening. I will be glad to take questions.
Thank you very much, Gilad.
Thanks, Gilad.
Yeah. So I think the floor is open for questions. Is Jyotishman, are you going to handle this? Because I don't see everything on my laptop.
Okay. Yeah. So, should it
You
be?
go ahead.
Yeah. So, there are a couple of questions. So, one I will let Anu Agarwal from BITS Pilani ask the first
I
question.
cannot hear you for some reason. Maybe I can.
Hello. Can you hear me?
Yeah, now I can hear you. Yes.
Okay. Can you unmute yourself and ask the question? Okay. Well, sorry, Gilad. There was probably a pause. So I'll let Advait Rishbad from IIT Bombay ask the question. Advait.
Hi, am I audible?
Yes.
Hi, thank you, Professor Haran, for your excellent talk. My question is with regards to the hidden Markov analysis that you spoke of. Since hidden Markov modeling is inherently an unsupervised learning technique, how do you validate the quality of your fitting once you have assigned states?
Oh, thank you very much. That's a very important question. Validation is something that we take a lot of care to carry out once we do the states. I don't have here slides to show you how to do that. There are several ways to do that. There are methods to actually take the data and what we call recolor it. In other words, assuming that we got certain rates, can we actually now go back and calculate what the data should have looked like and see if that's really reasonable. And if we get reasonable data, we take that as a good validation of the rates. We describe that in our papers. You can have a look there. But we have several different validation methods. That's a very important part of the line of work with this analysis.
Okay, thank you, Gilad.
Thank you.
Questions from the audience?
Well, Jyotishman,
yes, please.
I'm just, hi, Gilad, how are you? Arindam here.
Yeah, hi.
Hi. So I was kind of in the similar line of Advait. So imagine that you have during this fast folding that is happening, it's essentially these two domains coming close to each other, right? And you're saying that it's taking a few tens of microseconds at the most to kind of come close or take it out, right? So that's the process that you are looking at. Now, suppose I have a situation where the dynamics actually involves a third state, right? But it may not be that it's a dominant state. Maybe it's like has low probability with respect to the standard process. Would I be able to identify it?
Can you say that again?
So imagine I have a low probability situation where it actually, instead of two states that you mostly talked about, it goes through some sort of a transition state or some sort of an intermediate state with low probability events, right? How would you be able to identify it if it happens in this?
So the identification of states just depends first on the quality of the data, and second on our ability to analyze the data in terms of a model with n states. So I showed you just an example.
Two states.
We modeled two states, but we could do 3 states or 4 states or 1,000 states, right? But of course, 1,000 states, the data will not be able to give us anything.
That's right.
But
so what I'm
2 and 3, we can still get information about the 3 states at the same level, perhaps, as we get on 2. And by doing these validation states that I just mentioned, we can figure out whether that's a better description of the data than with 2 states. So, we go for the best description of the data. We always work on the statistical level. We have the best description statistically, and that's what we take as a proof that that's what the molecule has. So the answer is yes, we can get also a minority state if it exists there, if we put it into the analysis.
Okay. So the reason I was thinking about it is because when we have tried to do this solution measurement where a protein molecule is diffusing and then it's undergoing FRET or not, right? The moment we go below a certain time scale of acquisition, so you said you are working with 50 microsecond, right? Per bin, correct? Time bin. But we have found that the data becomes very noisy. Now, when I go to higher time scales, the data is cleaner, right? We can see the bursts cleaner. Is there any rule, thumb rule that we employ to, to get the best out of the situation?
So this is a very important question. It relates to the question of how many photons on the average you can collect from a molecule. So in a typical standard experiment, we don't collect more than a few tens of thousands of photons per second, which means that the burst, a typical burst is 1 millisecond. So in 1 millisecond, you cannot have more than a few tens of photons. Okay, that's the low side of the measurement, but it's very typical to see that. And so with a few tens of photons, the time resolution cannot be very high. But you can actually make an effort to increase dramatically the number of photons emitted by the molecule. There are several ways to do that. I don't want to get into how to do it, but imagine that instead of getting, I don't know, 20,000 photons per second, suddenly you have 200,000 photons per second. So you increase the time resolution by a factor of 10 by just increasing the flux of photons that you get. Now, very interestingly, in recent years, there have been a lot of work on how to increase the flux of photons from a molecule. For example, using, you're familiar with that, plasmonics. So you can actually create an interaction between a molecule and a plasmonic device. And because of this interaction, the molecule emits more photons. And so you can get suddenly, instead of a small number of photons, you get a very large number of photons. And this allows you to get a much better time resolution. We are working on that now. We hope to soon have that as a way to actually look at very fast conformational changes inside the proteins. But the idea is just look for the number of photons that you can get. If you get more photons, you have better time resolution.
Thank you so much.
Okay. Are there any further questions for Gilad? Okay, Shudiptada?
Yeah, no, I don't have anything. Thank you so much, Gilad. I really appreciate your giving the time amidst all that is going on, and hope to see you next month in Bombay. Okay, so thank you very much, and we'll wait for the next speaker.
Okay.
Gilad, thank you.
Big round of applause for Professor Gilad Haran.
Bye.
Bye.
Thank you.
Bye. Bye.
So we have, okay. I think Jorg is,
Jorg is going to set up the slides.
Mm-hmm.
Hi, Jorg, how are you?
Yeah, thank you.
Good to see you. Arindam here.
Yes, nice to see you. And I'm really overwhelmed how many people are here.
We have more than 110.
It's incredible.
Yeah.
So,
and I think we have more than a few hundred in the YouTube channel,
Yes,
right, Jyotisman?
50 or 60. Yeah.
Okay,
60.
around 50, 60
wow.
people in the YouTube.
So I will get famous.
Okay.
So can you see my slide?
Yes.
Yeah, you test your slides and start in a minute or two. The introduction, and then you can start. Check your slides.
Shall I start? No, okay, no.
No, just wait for so we'll wait for the 2-3 minutes. Yeah.
Yes.
I think for the students also taking continuous, a very intense talks can be difficult. So we give maybe a few minutes break for them and then come back between talks is a good idea. Jorg, how have you been? All good on your side?
Yeah, all good here in Göttingen. Yeah. So I'm looking forward to my visit to India.
Oh, yes.
I wasn't in India now for a really long time. I think last time before COVID. So it's really time to come back.
Yes, yes. We
Yeah,
look
very
forward
good.
to seeing you. I believe you were also here for the last Owls meeting, right? 2016
He was,
meeting.
yeah,
And
2016.
Yes.
yeah, that
yes.
was the time I actually got him for my lectures. He got a couple of special lectures on defocused imaging in my single molecule spectroscopy class in IIT Bombay.
Yes, that
So
was a nice experience.
yeah, that was a lot of fun. And I think right after that, one of the students went and did PhD with you. Arindam something, right?
Arindam Ghosh.
Ghosh, yes.
Wow, yeah, he was fantastic. He was
Yeah,
fantastic. He just got a science paper.
yeah. I talked to him last year. He was
He now switched to Australia, but I think he will be. He is outstanding.
Yeah.
Okay, I will start by introducing Joel. Audience, welcome to the next lecture in this series by Professor Jörg Enderlein. He comes from the George August University in Göttingen. Göttingen, of course, is the mecca of all recent science for the last 100 years. And there are new things that are happening, not just the Max Planck, but in the university. So I had the good opportunity to visit him a few years ago there and watch the fantastic stuff that is going on. Jörg has really been, if you talk about someone doing fluctuation spectroscopy, single molecule fluorescence, Jörg is probably the first name that will come up. And he has developed many recent techniques which are related to fluorescence correlation spectroscopy that some of us work on. So has really been a leader in that field for some time now. He also gives his time to the community. He is right now is the chief editor of Biophysical Reports, which is a new journal that is part of the Biophysical Society and is published by Cell Press. And he has been really generous with his time, because whenever we called him, usually he would say yes and come, and he has become a very good friend. And some of our own friends have gone and done stints with him, Manoj Kumbhakar, for example, and others who have also done very well in their own careers after these interactions. So Jörg has been a leader, and still he is blazing the path for fluorescence correlation spectroscopy. So Jörg, you're on.
Yeah, thank you very much for this very nice introduction. So I hope I can live up to the expectations. So I hope I did not make a mistake, because I was thinking I should present here something really more advanced concepts. I hope it's not too complicated, but let me simply start, and then, of course, you can ask any questions which may come up. So let's start with some very nice simple introduction. Of course, my topic, or my talk here, is about fluorescence correlation spectroscopy, and the main instrument that we use. This is the quite familiar confocal microscope, where we have an excitation laser, and then we reflect the excitation beam into our objective. We generate here basically a diffraction limited focus in our solution or our sample. Then we excite fluorescence here from fluorescent molecules, and fluorescence is collected by the same objective. It is passing the dichroic mirror, which is transmissive for the fluorescence light. And then we are focusing through the confocal pinhole to reject out of focus light, of course, that we also get a very nice restriction of the confocal volume along the optical axis. And then downstreams, typically we have at least 2 single photon avalanche diodes for detection, for high sensitive detection. And that's the main instrument what we use. And I guess this is quite familiar to everybody. So what is the basic idea of FCS? The basic idea is that when you now have a sample with a very dilute concentration of fluorescent molecules, which is shown here, so the yellow cigar should somehow visualize the detection volume. The diameter, as I said, is determined by the focusing of your laser light, which is roughly 1/2 of a micrometer. And the axial extent here is given by the confocal detection, which is then giving you a detection extent here around 1 to 2 micrometer. And then you have now a solution which is sufficiently diluted then what will happen is then when you take the fluorescence signal as a function of time, you will see a very strongly fluctuating signal, because all the time number of your molecules are changing inside your detection volume, and this is then giving this very strong fluctuation. And of course, when you come from physics, when you see such a fluctuating signal, the first thing which comes to your mind is to try an autocorrelation of the signal. And this is the basic idea of this fluorescence correlation spectroscopy. So we are calculating a second order autocorrelation curve from our fluctuating signal. And this is a typical example here. So the circles are data from the dye solution. It's a pure dye solution. What you see here are the different components of your correlation curve. Of course, at very long times, at very long delay times, we have a constant offset, which is uncorrelated light. Then the green thing here, this is the decay of the correlation due to diffusion, because let's say when you start with a molecule in the center of your focus, then with some time, this molecule will leave the focus and then you lose the correlation of the signal. This is the green curve here. And then we also see something very fast here on the microsecond time scale. And this is due to photophysics. I will not talk too much about it today. So this is, I guess, well known, and assuming as an example, of course, what you can do, or why people then invented it in the first place, is they wanted, for example, to measure the diffusion of molecules in solution. This is an example where we have here 4 different measurements. The blue one is a yellow fluorescent protein solution. The red one is the same thing, but now with dimers, so where we have 2 yellow fluorescent proteins covalently bound together. And you nicely see, yeah, the autocorrelation decay is shifted to longer times, which is then a signature that the molecules are bigger, diffusing more slowly, so the correlation decays more slowly. What we also did here, out of curiosity, we measured so-called pure buffer and water, but it's of course not pure, because you see something in the correlation analysis, which is contamination. This is also simply showing you how sensitive the method is. We are single molecule sensitive, so everything which is somehow fluorescing will contribute. However, I mean, this is all normalized here to 1. These green and blue curve are extremely dim. You have to measure long time, so it's much weaker signal than the red and the blue. And you only see it when there is no real sample in your solution. Okay, so far so good. My topic today is I want to show you some more advanced stuff what we can do. And the first thing is I want to talk about membrane diffusion. So I mean, this is what I show here. This is still in solution. That is what most people do, or let's say also when you go in the cytosol or look in cells. But what we did a few years ago, we started to be interested in membrane diffusion. And of course, you can use the same idea as of fluorescence as a correlation spectroscopy for membrane diffusion. Of course, the sample now is quite different from a solution. So you have to think about, okay, how can I prepare a lipid bilayer, a lipid membrane for doing my measurements? And there are 2 options. Most people, they use these so-called giant unilamellar vesicles. This is shown here. That's simply as a bright light image of such a big vesicle. Here the bar is here 20 micrometers, so this is something like a 30-40 micrometer diameter. And then you can focus here on one position on your membrane, and you can, for example, measure the lipid diffusion. Or what we are then doing in our lab, we use a more sophisticated system. I'll show you in a second, which is basically generating a freestanding lipid bilayer over this kind of Teflon membrane here. There's a Teflon film, and then there's a hole in the Teflon film, and we prepare a lipid bilayer on top of it. And there is a very nice commercial system. I mean, I have no commercial interest in this company, but it's a really nice system from the company Ionovation Explorer, or Ionovation, which is allowing us to do it in a very automatic way, and then also to be able to perfuse the upper and lower volume of this above and below the bilayer. And then we have full access from below with our optics, and we can even do then capacitance measurements and electrophysiology. And we used it in some years ago. We used it, for example, to ask the question, okay, how is the diffusion law in membranes? I mean, when you look in solution, then there's a very hard relationship between the diffusion coefficient and the size of a molecule, which is given by the Einstein-Stokes relation. I will show you, no, I will not show you today, but I mean, this is quite well known, this relation that the diffusion coefficient is inversely proportional to the size, to the radius of your molecules. The question we were asking, okay, what is going on when you have a lipid membrane? So what we did, we prepared our freestanding lipid bilayer, then we looked at the diffusion of the free lipids, then we had a very small single alpha chain protein here, the snare protein. We could then simply be labeled fluorescently and put it into membrane. Then we went to something bigger, which is a potassium channel. Then we had something really big, which is a alpha hemolysin. It's an exotoxin. From bacteria. Then we looked at the chloride channel, which is even bigger when you look at the diameter here in the membrane. And then the biggest thing that we looked there was a big drug pump from E. coli, the ACRB transporter. So we did then these kind of fluorescence correlation measurements and then determined the diffusion coefficient. This result is now shown here. I mean, it was of course a very long project over several years because it was not so easy to get all the biochemistry right. But what we then found is this blue relationship here. And there were two models at the time which were competing. The one was saying, yeah, even in the membrane, when you look at membrane diffusion, it is still governed by this Stokes-Einstein relation, which is for diffusion in solution. And there was from the 50s, there was a very simplified so-called Safman-Delbrück model, which was taking into account the membrane and viscosity differences between solution and membrane and so on, which predicted as a logarithmic 1/r relationship. And the point was, I mean, it was a big discussion at the time that it was not clear whether the super generic model, when you look at the derivation of this model by Safman and Delbrück, it is extremely generic and makes a lot of assumptions. So it was not clear whether this is indeed a true model, a correct model. And there were even reports that the Einstein-Stokes-Einstein relation would be the more appropriate model for describing diffusion in membranes. What we found is that the Safman-Delbrück model is extremely successful, which is shown here again. It depends on the viscosity eta m of the membrane, the viscosity of the fluid surrounding the membrane at eta f, the radius of your inclusion in the membrane, h is the thickness of your lipid bilayer, gamma is a constant. And T, of course, is the temperature, and Kb is the Boltzmann constant. So this is a very when you look at the derivation, it's really amazing that this model works so nice for real data, and we could really confirm over a large, how to say, spreading here of radius values that this model works amazingly well to describe diffusion of lipid inclusions in lipid membranes, which is quite important when you do biophysics. However, we not only wanted to look at the lateral diffusion of inclusions in the lipid membrane, I mean, we also wanted to see the membrane dynamics itself. So when you have a lipid membrane, it's very fluid, it's very flexible. So what you see in cells or in organelles is that the membrane itself can widely fluctuate spatially and also, of course, on a very fast time scale. So we wanted to see these very fast undulations, membrane undulations, again, using fluorescence correlation spectroscopy. But as I was demonstrate you now, that was not as simple as we thought in the beginning. So what we came up with is something, or we combined an FCS with something which is called graphene-induced energy transfer. And I have to make a very small detour to give you some idea what I'm talking about. So when you take a fluorescent molecule, then, and you want to describe its fluorescence emission, then this is extremely well described by the model of an oscillating electric dipole. So what is an oscillating electric dipole? You have a positive and negative charge. They oscillate along a given direction, which is the dipole orientation. There's an oscillation amplitude and frequency. Of course, when we talk about fluorescence, and this frequency are the optical frequencies. And when you put in this very simple model into classical Maxwell's equations, then you find the famous Hertz electric dipole emission, which is shown here. So this is the electric field distribution around your dipole emitter, which is in the very center here. Of course, the field is diverging in the model when you come to 0. That's why there is nothing shown here in the very middle. But this is the classical dipole emitter. And what you see, of course, you have a very non-uniform emission, which is described by a sine square theta law, where theta is the emission angle. I mean, of course, this wave is here with the speed of light, and then the oscillation frequency is something like 150 terahertz. What is important here for my next slide is that when you look very closely, this is quite interesting, there is no far field emission from such a dipole emitter along the axis of the dipole. There is no, when you would look from far away from above or from far away from below, you would not see any emission, because the emission is really concentrated in the equatorial plane and is going down to zero when you come to the polar axis. However, when you look very closely here in the very vicinity of your dipole emitter, then you see there is a non-negative electric field intensity. And this is, by the way, also the reason why we see first a resonance energy transfer, what was just mentioned by Gilad. So this is the idea that you have these non-zero field components here very close to your dipole. And when you now bring this dipole close to a surface, and let's say a non-trivial surface, which is here a glass substrate with a very thin metal layer. Very thin means really a few nanometer only, and then maybe a spacer layer, which is not so important here. When you bring your dye molecule, your emitter, very close to such a metal layer, then this near field, which I was just showing you, this near field can now interact with the substrate, in particular with the free electrons in the metal layer. And this is then coupling your dye molecule very closely to surface plasmons in the metal layer, and it leads to an increased de-excitation of your fluorescent molecule. This is very similar to what Gilad just mentioned in FRET, where you have an acceptor molecule. Here, we have only the donor dye, and the acceptor is the metal layer. And but in the end, you get a very similar process, that it's an increased de-excitation of your excited dye molecule, and this leads then to a strong modulation of the lifetime as a function of the distance. This is only happening very prominently in the near field region, which is here between 0 and 150 nanometers, so much below the wavelength of light. But in this region, you have this very strong decrease in lifetime when you come closer with your dye to the surface. It's also depending on the orientation of your dye. Fortunately, in most applications, the dye is rotating very quickly, so that we can use this random curve, the red curve here, for our experiments. The point now is the following. Because you have this very strong lifetime to distance dependency, we came to the idea, why not using a lifetime measurement then to determine the distance of a dye molecule from a surface. So if you can measure, let's say that your dye has a lifetime here of 0.6 in relative units, so the infinite lifetime value is 1. So let's say you measure a lifetime value of 0.6, then you can go here to this nice curve, and then you can say, yeah, that means my dye should be somewhere around 60 nanometers away from the surface. So that is the general idea, what we call the metal-induced energy transfer imaging. So we abbreviate here metal-induced energy transfer by MITE, M-I-E-T. So MITE imaging, so what we then invented many years ago was then to use indeed lifetime measurements of samples above a very thin gold film to get a very high resolution along the optical axis, and very quickly only. That was then the first paper in 10 years ago now. Basically, yeah, 10 years now, 10 years anniversary. And what you see here, for example, is the basal membrane of an epithelial cell, which is sitting and living on our gold-covered substrate. The cell is quite happy. It's the light cell imaging, and you see a time-lapse movie. And please pay now attention that the color here is indicating the distance above the surface. So it's the height profile of the basal membrane of the cell, and we have now a resolution of nanometers. So of course, it is very nice, but not yet enough when you come to a single bilayer. So if you want really to resolve single bilayer structures, and then in the next step, what we then basically realize is that you can use, instead of a metal, you can use something which is called graphene. So graphene is this hexagonal arrangement of carbon atoms. There was a Nobel Prize a few years ago for its invention, or for its discovery, better to say, in characterization. There's very unique properties, mechanical properties, optical properties, etc. But it can also act as a supergiant acceptor molecule for fluorescence. So you have again this very strong quenching when a dye comes close to the graphene surface. But now here, as you can see, on a much shorter length scale. So we have this from nearly 1 here to close to 0, the range is something like 30 nanometer. Again, it depends also on the dipole orientation. That is the red and the blue. And here in this graph, I also show you it also depends on the quantum yield of fluorescence emission of the dye. So two extreme cases here from 10% and 100% quantum yield. But this is all what also known a priori, so we can measure that is then very known. So we then exactly know what the correct curve is, but we have to use as the function between fluorescence lifetime and distance. And as an example to show you here how well it works, it is a single molecule data. So single molecules sitting on such a substrate at a given distance. So we use the 10, 15, and 20 nanometer spacer. These are single molecule fluorescence decay data in a logarithmic scale here. And you can see when we move our molecule only by 5 nanometers away from the surface, then we see a huge change in the fluorescence lifetime. And now let's come back to dynamics or to lipid bilayers. So why is it now so nice to use graphene? So we call it the graphene-induced energy transfer, because now we have such a high sensitivity along the optical axis, so conversion of lifetime into distance, that we were hopeful now to be able to even resolve the thickness of a single bilayer. So what we did is we started to prepare supported lipid bilayers on such a graphene substrate. We labeled the head groups in the upper and the lower leaflet, at the bottom and top leaflet. Then we did this kind of measurement. We determined the fluorescence lifetimes of the dyes here in the upper leaflet, the fluorescence lifetime in the dyes of the lower leaflet, and then we could calculate, this is shown here in the next table, we could then calculate the thickness of single lipid bilayers by a pure fluorescence lifetime measurement with no further, how to say, complications. So this is for DOPC lipid bilayer. This is DLPC. This is different chain lengths in the lipid acid chains. And then we found here, for example, for the DOPC, a distance between head groups of 5.5, here for DLPC of 4.04 nanometer, and really look at the accuracy. We are now really in the angstrom resolution regime. We also compared with small angle X-ray scattering measurements, which was a very good agreement. Our numbers are always a little bit bigger than the SAX measurements, because we have also an additional linker and fluor attached to the head group of the lipids. So the SAX measures really the head group to head group distance. We measure the fluor to fluor distance. But it was really very promising. And then we use it, for example, to look at lipid bilayer thickness modifications by cholesterol. So we have here the 3 different lipids here, 2 saturated lipids, 1 unsaturated lipids. We prepared then bilayers from these different lipid compositions here, and then we added more and more cholesterol, and then what we see is a swelling of the lipid bilayer thickness. Again, please pay attention to the resolution we achieve here now by a pure lifetime measurement. We are in the order of 5 angstrom, and we can nicely then see how my lipid bilayer is expanding by adding cholesterol. By the way, the unsaturated lipids, they show something like a phase transition. Yeah, first we have this expansion and then a contraction again. As a last example, then I come back to FCS, don't worry. But this is, I think I really love it. It was then done by my student Arindam Ghosh. I think a fantastic work together with Christoph Schmidt in Duke University and Sufi Raja. So they isolated single mitochondria intact, physiologically intact living mitochondria. They labeled again the inner membrane. It's a double bilayer system, as you may know. They labeled the inner as a membrane with one dye, the outer membrane with another dye. Again, we put it on the graphene sample. We measured the lifetimes, and then we could even do as in vivo measurements by then comparing what is the distance between inner and outer membrane of a mitochondrium when it's in the resting state, where you don't add any respiration buffer, and when we add respiration buffer, in this case pyruvate, malate, and ADP, so that this ATP synthesis machinery is switched on. And what we could see is that, of course, the position of the outer membrane is not changing because the mitochondrium is sitting on the surface, so there will be no change. But what we see is a change of something like 2.5 nanometer of the outer membrane, which is then coming closer to the inner membrane, coming closer to the outer membrane. And this is a live cell experiment, also basically a live organelle experiment. I would claim no other method at the moment could measure such subtle changes in a living system under normal conditions. So now let's come back to FCS. So why is it now an interesting idea to combine it with FCS? And it was developed, also everything I show you now, it was developed by my fantastic postdoc, Tao Chen, here in our lab. So he said, why not combine now this high resolution, Excel resolution of a graphene-induced energy transfer with fluorescence correlation spectroscopy, to combine the high temporal resolution of FCS with the extreme spatial resolution of graphene-induced energy transfer. And the first example, what he tried out was to prepare again these giant unilamellar vesicles, but now in such a way that they are basically attaching partially to the surface, simply by hydrophobic interaction. And this is shown here. Here's the rim of the GUV, and this is basically the contact area between the GUV and our substrate. And then he looked from the bottom, so he had the substrate, also he compared different substrates. For example, when we only have glass, no graphene, nothing, then we would not see any intensity fluctuations, because I mean, even when your membrane is here fluctuating up and down in the focus is gigantic. The focus diameter here, the confocal volume is something like 1 to 2 micrometer. The fluctuations we expect are only a few nanometer. So when you have this bilayer fluctuating up and down, you would not see anything in a normal standard FCS experiment. This is shown here. Even when you defocus, so when you shift your focus really below the membrane so that you get higher, that you expect higher intensity changes when the membrane is fluctuating up and down, you don't see barely nothing. However, when we now have our graphene, or in this case it was a metal, again a gold film, when you do this kind of giant trick, then we induce huge intensity fluctuations, because when the membrane moves up, then the fluorescence comes up. When the membrane moves down, fluorescence is quenched more. And this is inducing very strong intensity fluctuations, which are now clearly seen here in the fluorescence correlation curve. So that means you can indeed now very nicely see tiny membrane fluctuations. And what we did is then we used here these experiments, to comparing then GUVs in different states. So where we have a very high, where we have a very high osmolarity difference between inside outside. So then it's a very high tension. In that case, you don't see much membrane fluctuations, and then we have a very small osmolarity difference. Then we have basically a deflated GUV, which is very flexible, very strongly fluctuating. And from these measurements, we then can extract something like the surface tension or the interaction strength between membrane and surface. This is shown here. And of course, it was in vivo in vitro for GUVs, but of course, the more interesting thing is, can we also do some interesting biophysics with it? And the next thing what then Tao did, he looked at membrane fluctuations of red blood cells. So this is shown here. So you take individual red blood cells, again, you label the membrane with a fluorescent dye, you put it on the surface. These blood cells, they have this very special shape. So what you see here is then only the rim where the red blood cell sits on the surface. This is then shown here as a lifetime image. And again, then using this lifetime or intensity information, you can then again do FCS measurement here. And for example, then compare what is happening is when we add ATP for energy production, or when we deplete it with ATP in our solution, so that we reduce basically the energy production in the blood cell. And then we can extract something like the fluctuation, size. In this case, what you see here, this is the sigma of the fluctuation is in the order of 6 to 10 nanometers, and also we get the fluctuation dynamics. So this is the correlation time, the characteristic correlation time when the correlation has fallen off to 50%. You can even do it with graphene. It was all with gold. Now with graphene, then Tao again did a fantastic job to prepare very special substrates where he had these kind of 1 to 2 micrometer pores with very shallow depth here of only a few nanometers. And he spent a freestanding lipid bilayer to get away from the solid surface interaction of the bilayer. You see here these pores. These are different pores on the graphene substrate. Some of them are dark. The bilayer has ruptured, so there is now no signal. But then here, these, for example, these pores, they have a spanning bilayer, and again, you can measure then the fluctuation of these bilayers. And for example, in this case, it's only 1.3 nanometer fluctuation sigma, and with a fluctuation time, a typical fluctuation time of a few milliseconds. And again, then we went on to look at the living systems. Again, we have chosen mitochondria on these graphene samples. What you see here is the lifetime image for the outer membrane, which is more bluish because lifetime is shorter. Here's the inner membrane, more reddish because lifetime is higher. And again, we can then use basically FCS to look at the fluctuations and then compare, for example, here when we add again ADP to our mitochondria, or we deplete our solution with ADP, and then, for example, you can then look what is happening with your membrane dynamics, and mostly nothing so much changes. However, the membrane fluctuation spread is getting increased when you add energy sources here, ADP plus. Then the fluctuation will get stronger, and also the time becomes much shorter. So that makes when you add basically something for respiration, then these mitochondria become much more dynamic, the inner membrane. So it's a highly dynamic system. Yeah, so back to the next concept. I mean, I talked already a lot about lifetime, so how to use basically, I mean, that was somehow also inherent in these measurements, some lifetime measurements together with FCS. But there is one concept we worked out also many years ago, and I think this is quite interesting in the context of fluorescence correlation spectroscopy, which is called fluorescence lifetime correlation spectroscopy. So give me 1 second simply to explain a little bit more about how we measure lifetime. So I was talking all the time lifetime measurements, but how we do it. So we use a pulse excitation. So we are exciting our fluorescence with a pulsed laser, which has a high repetition rate on the order of 25 to 80 megahertz. And with very short pulses, sub 100 picosecond, 50 picosecond pulse width. And then what you do is you repeatedly excite your sample, and then you time the delay between your fluorescence photon detection and your last excitation pulse. And then you histogram these data, then you see this very nice, and I guess everybody of you knows that, this very nice fluorescence decay curve. So this is the idea. So what we then did is we combined these lifetime measurements with fluorescence correlation, and this is a little bit complicated to explain. Let's assume we have the following thought experiment. We have a solution where we have a mixture of two components, A and B, and the only difference between these two components is their fluorescence lifetime. Otherwise, they have the same excitation spectra, the same emission spectra, etc. But their lifetime is different. So A, let's say, is a short lifetime component, and B is a long lifetime component. So if you only measure intensity fluctuations with a standard fluorescence correlation spectroscopy instrument, you would see something, in the best case, you see something like that. Some broad decay of your correlation curve, of course, which is determined by the diffusion of these two components. And if they would have the same diffusion coefficient, then you would see simply a standard normal single decay FCS curve. So what we wanted to come up with is a method that we can indeed, in an FCS experiment, distinguish between these two components. And what we did is the following idea. So what you do is you are finding so-called filter functions. This is blue and a red curve here. In the nanosecond timescale, here is the same timescale as the fluorescence decay curve. And these filter functions have the following property. If you take the blue decay curve here, this is the blue curve, what you would see, let's say, when you do this pulse excitation. And you multiply this blue curve point by point with the red filter function here. The result would, when you sum then over all these multiplications, the sum would be 0. So that means this function, the blue function here and the red function here, they are orthogonal to each other. When you multiply the red curve here, the long decay curve, with the blue filter function here, again, you would see they are orthogonal to each other. And then you do it pointwise and then sum up, it would be 0. However, when you multiply the blue curve with the blue curve, you would get unity. And the same thing with the red curve. To the red curve, you would get unity. So what we do is, before doing a standard correlation calculation here, the two-photon correlation, we are weighting every photon with either the blue or the red filter curve here. And only after that, we calculate correlation curves. And when you do it, then you can, in general, you can generate 4 correlation curves from one measurement. So here I only shown 2 of them. This is the autocorrelation where you correlate, let's say, the long lifetime photons against the long lifetime photons, and here the short lifetime photons against the short lifetime photons. But this is the idea of this fluorescence lifetime correlation, that you use the lifetime information in the same way as people use the color, let's say, spectral color in fluorescence cross-correlation spectroscopy. And there's a very nice example, again, my fantastic former student, Arindam Ghosh. And it was one of his projects. So he used it to elucidate the photophysical dynamics of green fluorescent protein. When you take green fluorescent protein, GFP, and you look at the lifetime, then you will see very quickly that the lifetime is not mono-exponential, but is a bi-exponential decay. And of course, the question is, what is the reason of that? Does it mean that the maturation is incomplete? So some of the GFPs, they have a short lifetime, and the more mature GFPs have a long lifetime? Or does it signify that there is some really intrinsic dynamic lifetime dynamics within one and the same GFP? So what he did is, he applied now fluorescence correlation, lifetime correlation spectroscopy to the data from GFP. So you have a GFP solution, you identify the 2 lifetime components, you use then these 2 lifetime components then to do an FLCS experiment where you can then basically calculate here the autocorrelation of long lifetime photons against long lifetime photons, of short lifetime photons against short lifetime photons, but also, this is the interesting part here, of, let's say, long against short and short against long lifetime photons. So this is something like a cross correlation across different lifetime values. And what you see here now is something very exciting. You see this kind of anti-correlation, which is signifying that when your molecule starts in a short lifetime mode, that with some time, and here in this time scale, as you see, it's in microseconds, then after a few microseconds, there's a good chance that your molecule is switching into a long lifetime mode, or vice versa. And from this kind of fitting here, these data, you can get these transition times between the lifetime states. And it was a very nice then proof that what we see, that the bi-exponential decay in GFP fluorescence is indeed an intrinsic fluctuation of the conformation of the GFP, and we could then even correlate it with X-ray data, which are showing that indeed there is a flip-flop of one amino acid close to the chromatic center of the GFP. But such a measurement is only basically doable with this kind of method. Of course, again, let's come back to the membrane. Then Tao and Arindam, they joined forces and said, oh wow, when FCS is working so nicely, why not combine it again with my graphene-induced energy transfer lipid bilayer stuff? So what they did is they prepared lipid bilayers here on a graphene substrate, they labeled again, and then they added cholesterol. They labeled the cholesterol with a fluorescent dye. And the idea was the following: it is well known that cholesterol can do a flip-flop, or lipids generally can do flip-flop between leaflets, so that let's say when here cholesterol is the beginning in the top leaflet, and it can switch to the bottom leaflet and vice versa. But to measure this flip-flop dynamics is quite challenging. So the idea was, okay, now with graphene, which is inducing a very strong quenching, distance-dependent quenching, and then combining it with FCS measurements, maybe we can then, and then also using FLCS, a lifetime correlation, maybe we can then really use that all together to get the dynamics of this flip-flop. And indeed, it works. So this is the fluorescence lifetime correlation spectrogram of the cholesterol, of the lipid, also of the labeled cholesterol in a lipid bilayer on graphene. And what you see here again is the autocorrelation of long lifetime against long lifetime, short against short lifetime, and here's the cross correlation again. The anti-correlation here, this is telling us how fast the flip-flop is taking place, and we find values here super fast in the millisecond to sub-millisecond time range. Very surprising because I think it was not seen really before, and we are still struggling to understand whether this is an artifact from our sample preparation or indeed that is the real flip-flop dynamics. So we are still in the middle of confirming that. But I simply wanted to show you that this method can be extremely powerful and promising for such kind of studies. So let me simply then use the last remaining, I guess, 10 minutes of my talk to come to one of my beloved topics, which is not exactly FCS, but is very closely related to FCS. This is stochastic or super resolution optical fluctuation microscopy, and you will see in a minute why there is a very deep connection to fluorescence correlation spectroscopy. So this is now delving into the realm of super resolution microscopy, and I guess you are all familiar with these famous methods of photoactivation localization microscopy, PALM, or stochastic optical reconstruction microscopy, STORM, which were basically the same time invented by Eric Betzig and Xiaowei Xuan. Then Eric got a Nobel Prize. Xiaowei mysteriously did not get a Nobel Prize. I don't know why, but it was basically the same idea. And the idea was, okay, that when we want to do super resolution microscopy, what we can do is we are labeling our samples with fluorescent dyes, and then we have some kind of magic to switch off of most of the dyes into a dark state. So then we take a single fluorescence image that we only see a very few of our molecules in the bright state, which is shown here. And then the idea is, this is the core idea of the whole method, that if you know that at a given spot here, there is only a single molecule, or so the width of the image of this molecule is given by the resolution limit of your microscope, and typically is in the order of some hundreds nanometers. If you know there is only a single molecule, then you can localize this image with a much, much higher accuracy than the width of the image. That was the idea. So that means you switch off most of the molecules, you switch on only very few of them, you localize them, then you switch another set on, you localize them, you do it over and over again. In the end, you have a huge list of single molecule coordinates with an extremely high accuracy, and then this gives you then the final image. But this is a different story. What I want to show you today is, again, a clever idea coming from fluorescence correlation spectroscopy. Because in these kind of methods, PALM, STORM, the temporal information, how these molecules switch on and off, is completely neglected. That's only a means for the goal. What people want to do is simply they want to simply switch on and off different molecules in different images. So we were thinking, maybe we can use ideas from FCS to also improve resolution in a simpler way, which is not requiring this very complicated switching and this localization business. And this is going back now a little bit of mathematics. So we have to ask ourselves, okay, how is the image on our camera formed at any time, a given time? So T is the time where we take the snapshot. X should be the position on our camera, so it's the pixel position on the camera. Of course, in reality, it's a two-dimensional coordinate. And then what you see on the right-hand side is the sum over all fluorescent molecules in your samples. Sk should be the intensity of your molecule number K at a given time T. This is then the contribution of this molecule to your final image. And then, of course, the image is formed with the so-called point spread function, which is shown here, which is telling you, okay, if my molecule K is located at position xk prime in my sample, so what is then the contribution of this molecule to the fluorescence signal recorded at position x on my camera? This is the point spread function. So it's a linear relationship, very nice and very easy. So this is the image formation. Of course, the width of this function here, the width of this function is determining the resolution limit of your normal microscope. So when you have a very dense labeling, then this U here, the width of this U, is determining how good you can resolve something in your sample. So far, so good. Now comes again the correlation business. So we are back now to fluorescence correlation. So what we were thinking was the following idea. So let's look only, let's say, on two different molecules, J and K. and only on their fluorescence brightness, intensity. So that is the only thing. We are completely ignoring at the moment their positions in the sample. You only look at the fluorescence brightness, let's say, of molecule J and molecule K here, 2 molecules. And what we do is we then do a normal, how to say, a 2-photon correlation analysis. So we are subtracting the mean intensity of the molecules as J and as K, this is here with this bar, and then we form the product, and then we average over all possible starting times T naught here. So this is what, when you are familiar with FCS, it is basically the definition of a 2-photon correlation function, really, that now here the signal comes from molecule J and this comes from molecule K. So the basic idea is the following. Let's say your molecules are fluctuating, intensity fluctuating. Normally, and this is what we see in many, many cases. So many emitters, they have intrinsic intensity fluctuations. However, if these molecules are not physically interacting with each other, then these intensity fluctuations are non-correlated across each other. That means when J and K are different numbers here, then the result will be 0. Yeah, when you correlate, let's say, fluorescence intensity for molecule J with the fluorescence intensity for molecule K, and from different molecules, the result will be 0, which is shown here by this Kronecker symbol. Only in case when you correlate the signal from one and the same molecule, so J is equal to K, then you will expect the standard fluorescence autocorrelation of the fluorescence signal from this one single molecule. And the funny thing is, you can extend this idea to higher orders. This is then called not correlations, but cumulants. So for example, third order cumulant looks like this. And again, you have here, let's say, 3 different molecules, J, K, L. And only when J is equal to K is equal to L, only in that case, there is something non-zero on the right-hand side. This is then the free photon cumulant function of your fluorescence signal from this given molecule. And you can generalize that to arbitrary order. So why is it now interesting for imaging? So now let's again take our image. It was here our image. That is a function of this X, and the sum here runs over all molecules. And what I'm now doing here, I simply blindly apply this kind of second order correlation operation to my image, so pointwise. So I have here X, here is the same X, but now I simply do this kind of temporal correlation for every pixel in my image. This is shown here by this expression here. And of course, then this double sum can be rewritten, as simply rewriting into this form here. And this averaging here, these angular brackets over time, this is only then applying to the fluorescence intensity of my molecules. This is shown here. And again, think about, then J is not equal K. So when we correlate here fluorescence signals from different molecules, then the result is simply 0, because fluorescence fluctuations between different molecules are not correlated. Only if J is equal to K, then this is a non-zero function. So it means the result here becomes then something like this. This double sum is collapsing to a single sum, where this function F2 is in the second order autocorrelation of the signal of one molecule. And here this product is collapsing to the square of my Poinsford function. And this is super interesting, because this is again something like an image. It's depending still here on the position X on my CCD camera. So, but now this kind of artificial image, which I have now calculated, is formed with the square of the Poinsford function. And when you think about it, when you start with a Gaussian of some sigma, then the square of this function has, of course, a smaller sigma. So that means this image here has an effective Poinsford function, which is smaller than the original one. You can generalize that by using higher order cumulants. Mathematics is a little bit involved. Not so important here, but I simply show you the equation. So let's go to practical applications. So how do we do it? So what we do is we take simply our sample where we have nicely labeled emitters, and then these emitters are intrinsically fluctuating. This is shown here as a kind of, this is a calculated example. This is a movie. So we take a movie, and then by pixel, we do, let's say, in a classical second order correlation function calculation. We calculate the autocorrelation for every pixel in our image. And then we take the amplitude of the autocorrelation as the amplitude of the new image. This is shown here. And when you do that, you will then find the very nice final image. And indeed, and believe me, the resolution here now is by a square root of 2 better than the resolution of the original image, because you have now formed your image here with the square of the point-search function of your original microscope. And you can now extend it to arbitrary high orders. In the very first paper in 2009, together with Shimon Weiss from UCLA, what we did is we used quantum dots, which are basically indestructible. We did this kind of analysis, and you see here, this is the raw image. That is the second order of SOPHY correlation, fourth order, 16, 25. And here are cross sections. And indeed, by simply going to higher and higher orders, you can basically, also ideally, you can squeeze your resolution to arbitrarily small numbers by doing a simple time correlation of your movie data. That is really some kind of magic. Of course, in reality, we are limited to something like roughly fourth order, because it's otherwise you need extremely long measurement times to get a reasonable signal. So how does it work? That is again from this paper. So for example, here it was a tubular network in a fibroblast. We labeled it with quantum dots, nicely intrinsically fluctuating widely. You can also see the label density is so high that there is no chance to see single molecules here. You cannot do single molecule localization anymore. However, you can easily now apply the SOPHY trick. The result looks like that. And there are two things to mention here. The first thing, indeed, we have a resolution enhancement. Believe me, that is basically doubled here from left to right. But also what you see is that all this haze here, all this background is gone, because everything which is not fluctuating in time is completely canceled. So that means autofluorescence, scattering light, whatnot. So this is also one big advantage of SOPHY, that you can get extremely high contrast images by simply canceling everything non-fluctuating. There was one technical issue I want to mention very briefly. This was the following thing. When you take an image, let's say with a normal standard CCD and a normal fluorescence microscope, then typically you adopt your pixel size so that the effective pixel size in your sample is roughly something like half the point-search function size, so 100 nanometer. Now I told you just, yeah, but applying something like a fourth order SOPHY, you can 4 times getting better resolution. But how can it work when your pixel size is still 100 nanometer? So when you do SOPHY on something like that, then you get some really very coarse-grained final image. So the idea we came up, I think this is very quickly to mention, is again quite simple. And what we simply realize is the following thing: when you take your raw images from your movie and you do
George, time is up. So if you can wrap up in a couple of minutes, please.
Okay, then let me simply wrap up here. This Fourier upsampling is not super important. Let me simply show you some last things here. Yeah, I mean, I can also upload my slides then for when people want it. Let me simply mention one thing. One really nice final is also a property of SOPHY is that you have three-dimensional sectioning in wide field images. I mean, what you do is with SOPHY, you take wide field images as a movie of wide field images. If you would take wide field images and you would stack them together for getting three-dimensional information, it would not work. However, SOPHY has, again, by this correlation idea, the intrinsic sectioning capability, which is shown here. And I think this is the third dimension, as a third property of SOPHY I want to mention here. I have also some other biological data here. Let me simply finish with this final image here of a mitochondrial network in 3D, which is quasi-simultaneous 3D imaging with SOPHY. That was published again 10 years ago. And so look it up if you want to see that. And I have to thank my whole group here in Göttingen and of course all the funding. And I hope it was not too much information today. Thank you so much.
Well, thank you. Okay. Big round of applause for Professor Enderlein for telling us about what all are possible with a little bit of fluctuation, and especially when it is helped by a little bit of a metal or graphene and things, and you get to 1 nanometer kind of resolution. So fantastic. The good lecture is something when you hear it, you think that why am I not doing it? And that's what I was thinking listening to Joel. So I have, of course, many questions, but I'll first go around and see if there are other people asking questions, and then we'll come back to my questions.
Yeah. Okay. Okay,
Jyotish.
Shudipto, there are 2 questions. One from Pramod Kumar. I think, Pramod, can you unmute and ask the question? Okay, since Pramod is not able to, I'll just read out the question here. I have two simple questions. The first one is, how do you prepare your lipid sample to keep on the top of the objective lens? One. And the second one is, in your shown data, you only used one type of lipid system, that is zwitterionic lipid. So my question is that, have you tried measurements for anionic or cationic lipids?
No, we didn't. I have to say, because the preparation of these lipid bilayers is a little bit more complicated, and but in principle it would be possible. So I mean, one has to do more tricks, I guess, to get a stable bilayer. So for doing bilayer measurement as a preparation, we use different methods. One, as I said, was this kind of spreading a bilayer over a Teflon pore for getting a freestanding bilayer. It was also used by Tao for preparing freestanding bilayers over these nanopores I have shown before. And this is basically by something which is called painting. So we are painting bilayers over these pores, and with some little exercise, it's not so super complicated. Otherwise, when we do the supported lipid bilayer, we use a Langmuir blotched truth technique. This is quite standard, so it's a commercial instrument where you dip your sample into a solution, where you have on the surface the lipids swimming, and then by simply dipping in and out, you get a very highly ordered and very well-defined bilayer.
Yeah. Okay, sorry, that was an uncorrelated noise. Sorry about that. So, yeah, please go ahead. Yeah.
Yeah, no, that was the answer, I hope.
Yeah, okay, answer. Yeah. And the second question from the chat box is Arun Kumar Upadhyay. He's asking, how can we explain the correlation if our fluorophore have different sizes with different lifetime that we are not able to separate out?
Also, I mean, that was only an example. So, I mean, but the fluorescence lifetime correlation is very useful in cases when everything else, for example, diffusion speed, excitation, emission spectrum, and everything is identical. In one example, I think it was as one of my favorite examples, was a GFP sample. You have a sample, yeah, in this case it was GFP. You see there are 2 decay times, but you have no idea, is it dynamic, is it static, what is the reason? So, for example, when you see 2 decay times, it could be that you have 2 subspecies. Yeah, subspecies 1 with short decay time, subspecies 2 with long decay time, but you cannot learn anything from standard FCS about any dynamics which is going on. So with FCS, you can really resolve that there are transitions between the lifetime states. You see the dynamics. And this is, I would say, that's the only method how to do it. No?
Okay. Thank you. So now the floor is open for questions from other people. Shudipto, you can.
Okay, so Jörg, I have 2 questions. One sort of a technical one, and one about the sort of the future. So the technical one is, when you look at the membrane correlation, the way we derive the typical equation for FCS assumes that at t equal to 0, your concentrations at some r and concentration at R prime are not related. They're not correlated at t equal to 0. And diffusion sort of connects them across time. But when you take a membrane which is fluctuating, across your probe volume, things are fluctuating in kind of a coordinated fashion, unless the fluctuation spatial scales are very small. So would the equation really work to fit?
Naja, also, okay, good question. Also I mean, you have to, when you apply our graphene-induced energy transfer in FCS to membrane, then you have basically 2 different processes. One is the lateral diffusion of lipids, or any inclusion you have labeled in the membrane is a lateral diffusion. This is also what you normally see when you do FCS on membranes. But now the second component, or the second process, is the vertical undulation of the whole membrane. Interestingly, the time scales are quite different. I mean, typically the diffusion time for lateral diffusion is order of a few milliseconds, and the undulation time of, let's say, for example, these giant unilamellar vesicles on the microsecond time scale. So there's very strong division. One interesting question, maybe what you are asking, is whether there's a correlation between undulation and lateral diffusion. very interesting, we did not see that. And I guess the reason is that our lateral diffusion is so much slower than the vertical undulation. So that on the time scale of the lateral diffusion, at least the lateral diffusion through the confocal volume, the vertical undulations are completely averaged out. Yeah.
Okay, thank you. The question about the future is, and it's all great that we look at these and derive equations that can analyze the data and we understand the parameters. But there is an orthogonal approach that's coming in, which is just look at the photon statistics and use some sort of the Bayesian sort of the approach to extract data that doesn't lead you to some equations or parameters, but can in principle extract more things than these equations can. We'll all be together a month later with Steve Press, and he does some of that. There are other people doing it also. Any comments about that? Where are we going from here in FCS?
Yeah, good question. I mean, the problem, I mean, I'm big fan of the approach of Steve Pressé for sure. No? The problem is you need tons of data for doing this kind of analysis. So my experience, only my experience, I can tell you, for many years ago, we tried to also use higher order correlation functions to get more information. So 3-point, also 3-photon correlation, 4-photon correlation. What we had to learn the hard way, the higher you go in your correlation order, the measurement time needed for getting sufficient data is exploding. Yeah. And in the end, it was not, at least our attempts were not so super successful. We never really could achieve enough data to extract useful data from higher order correlation. And I think the similar message, I guess, would also be told by Joachim Müller, who did very, very good work on higher order correlation functions some 10 years ago in the US. And also it was not so successful. I know that Steve now tries to use this full Bayesian approach to get much more information. But again, I think the limitation in practice is then the photo numbers you can extract from your measurements. Yeah. And that is the biggest bottleneck. Yeah. So in the end, at least here, we are trying to keep it as simple as possible.
Okay, good. So thank you. We're really looking forward to having you here in a month.
Sorry,
and,
Sudipta, can I?
Yeah,
There's one more
you have a
question.
question.
There's one more
I
question.
was just waiting.
Hmm.
I am actually asking a question. This question, George, is a very lovely, lovely talk. I learned so much about the last part when you talked about the fluctuations.
Yeah.
I don't know if you're aware, quite some time back, there was a paper published in Journal of Physical Chemistry about two quantum dots which are separated by more than, I believe, around maybe half a micron away, okay? Which showed some sort of a correlated blinking, okay?
Yeah.
And it was attributed to like the emission triggering the emission of the other. Now, I don't really believe it, but do you think that any of those effects can possibly influence your analysis?
Yeah. Oh, yeah. I mean, absolutely, yes. I mean, the SOFI concept is relying on the assumption that the fluctuations of different emitters is completely statistically independent.
Independent, isn't it?
Yeah. However, I have to say, I would have big pain to explain how the two quantum dots, which have a distance of, let's say, 2 wavelengths apart from each other, how they could interact with each other. Because, I mean, you could think about something like FRET, but you know how quickly FRET is decaying away with this.
Yeah, it's. I will share the paper with you. Maybe once you come, we can discuss. And I also have some data to share with you. So
yeah, that would be extremely interesting. But of course, something like that would destroy our basic assumption. Yeah. Fortunately,
no,
I mean,
I just remembered that paper, and I never understood that paper. So I thought that you might comment on that.
Yeah. No. Of course, it would be a big trouble. For Sophie, for sure. No? But I mean, but we never, also we looked into that, but we never saw any cross correlation between emitters, which would be hard to explain physically, I would say.
That's right. Thank you. Thanks so much.
Okay, one final round of applause for Joel. Thank you so much. And we see you here in a month.
Yeah,
Okay.
I'm
All
happy
right.
to come. Okay.
Bye.
Bye.
Bye till then.
Bye-bye. Thank you. Bye.
Bye. And to the audience, we have a break now, exactly 1 hour.
Yeah.
And one of the real stalwarts of single molecule imaging and spectroscopy, Taek Ha, would be waking up in the US and delivering a lecture. At 5 o'clock. As for all the other lectures, join 10 minutes in advance and that'll be the last lecture of this fantastic day. Okay, so see you at 4:50. Bye.
Thank you. Some of you can continue in the Zoom. Thank you.
Some of them can just be here without. So I was wondering, JT, I mean, TJ Ha is really the stalwart of single molecule FRET, right? He's one of the real stalwarts. But his talk title I saw is about
Mechanotransduction at single molecule level, actually.
So he has like moved on to some advance. I don't even know what that is. Look forward.
Yeah.
ठीक है चलो.
I hope he makes it. I hope he makes it to the meeting next month. So
he is coming, right?
Yeah, he's coming. Yes.
I am more worried about Wahid. So
yeah, we'll see. Yeah.
Chalo, I'll talk to you soon. Bye-bye.
Has TJ joined yet, Jyotishman?
Okay. I don't see.
Not in the waiting room, right?
No.
Okay, there's still time.
Maybe send him a reminder. Only 10, 11 minutes are left.
He sent something this morning saying that he's okay with recording on YouTube. So he remembered then. So I'm sure this morning means not my morning, his morning. That is a couple of hours ago. So it should be fine. Anyway, I told him 5 minutes early would be okay. So should be fine. Let me just see if he has any other message.
Fine.
Uh,
Waiting room, there are two people in the waiting room. Mansi Kothari and Gitanjali. Hey, we have
Gitanjali,
to
I
send
know.
it.
Okay.
Gitanjali,
At 3:05 our time, he sent me a message. So should be fine.
I think we'll just wait. Hello? Eh?
There's somebody named Mansi Kothari has been waiting in the waiting room for a long time.
I know, but
Let it be, because she has been there all day. And I think she
Oh.
has joined using another name with the institute anyway.
I see.
So let it be.
Okay, fine.
मैं तो स्पीकर के लिए वेट कर रहा हूँ भाई.
Zoom participation is still good, though, of course, waned a little bit from the morning. I'm sure more people will join in by 5 o'clock. Okay, maybe I start by introducing Professor T.J. Ha. So, audience, friends, colleagues, welcome to the last session of today, the evening session. We have Professor Taek-Chip Ha, who is a Bloomberg Distinguished Professor in Johns Hopkins, but actually very recently he has another prestigious professorship from Harvard University, and that's where he has moved. So if you look at his career, if I just start talking about the prizes, there'll be no end to it. I can read out a few. So the Searle Scholarship, the NSF Career Award, the APS Award, Alfred P. Sloan Foundation Fellow, Howard Hughes Fellowship, which is basically they say you're a member of the Howard Hughes Institute, only the very best get it. And finally, the membership of the National Academy. There is TJ. Okay. Welcome, TJ. Welcome to our evening session, your early morning session. Can you hear me?
Yeah, hello. Can you hear me?
Yes, absolutely. Very good. Very good to have you here. And I started introducing you, and my job is almost done. So, so instead of talking about awards, etc., what I'll say is that from the last about 20 years or so, all of us who are doing imaging, single molecule, etc., have to, as a matter of religion, almost read his papers every time something new comes up. And of course, much of it came out when he was, so I think about 15 years or so, he was at UIUC, University of Illinois at Urbana-Champaign. But then he moved with some distinguished professorship to Johns Hopkins. And as I was telling them, I think you have left that and you are in Harvard now. Are you holding both positions? TJ?
Yes, yeah.
Okay.
Also, yeah, I'm a serial mover.
Okay, all right. So he has moved then. That's mistake. Your Wikipedia profile still says that you are at Johns Hopkins.
Oh, I
changed.
know, but I never had anything to do with my Wikipedia profile, so
I see.
I guess.
Okay, so we are very, very glad to have you, and even more excited to know that this interaction will finally get to an in-person interaction in about a month. And Professor Ha is the known single molecule expert, imaging, etc. And one of our own closer circle, Rahul Roy, worked with him as to get his PhD. That has been also, I think, 10, 15 years. And thank you so much for appearing for this and also for agreeing to come in a month. So I think it's about 2 more minutes left. Okay, so we'll start in 2 minutes. You want to put your slides up and see that if it's working?
Yes.
Please. Great, that works. Yeah, sorry for this early morning intervention, and thanks for agreeing to do it. So, yeah, in about a minute, we can start. So it looks good. And I think if there are videos, you might want to just check them out. If there are no videos, then they should be fine.
I have some videos, but that should be fine.
They should be fine. Okay. Okay, TJ, you're on.
Okay, thank you very much for the invitation and for the opportunity. It's really great honor and privilege to give this talk in advance of the exciting meeting in Mumbai. I really look forward to it. Actually, I'm coming with my wife, who is also a speaker, and also we were able to convince our youngest in high school to skip a week of school to join us. So that'll be really fun. So my name is Taek-Chip Ha. I go by TJ. I work in the program in cellular and molecular medicine at Boston Children's Hospital. And we also are part of Harvard Medical School. And I'm also an employee of Howard Hughes Medical Institute. So today I will give this lecture that I picked this title, Mechanical Transduction at Single Molecule Resolution. So beauty and majesty of life is all around us, starting from the tallest tree in the forest and hummingbird hovering over a flower and down to the smallest insects. But if you zoom in, the life continues to occur to ever smaller scales into the cells and single molecules inside the cells. So the big challenge in understanding life is to really deal with vast scales of space, time, and complexity. We think that biophysics is very important in this respect. In fact, if you look at this vision statement from the Biophysical Society that we drafted 3 years ago, we want to harness the full potential of biophysics to seek knowledge, improve the human condition, and preserve the planet for future generations. In fact, many of the discoveries made in terms of advancing biophysical technologies have been honored by the Nobel Prizes in various subjects. Of course, the most recent being the protein structure prediction. To highlight two important discoveries related to single molecule experiments, biophysics. One is super resolution fluorescence microscopy, and the other is optical tweezers and their applications to biological systems. So let's look at optical tweezers. So I call them chopsticks that made of light. So you use a focused laser beam to grab a small particle or bead, and then you can apply very small forces down to a piconewton of force. And against, for example, an enzyme sitting on a glass surface that is transcribing DNA, producing RNA, and then by playing a tug of force with tug of war between the optical tweezers and the enzyme, we can actually gain information about the enzyme kinetic steps and mechanisms at extremely high resolution. And in fact, the idea that you can extract information by stretching mechanically is not new. Throughout the human history, we have learned that information can be extracted by stretching. And for example, you can ask how flexible is DNA using optical tweezers and gain additional information about that topic. So my lab is best known for our work on single molecule FRET for fluorescence resonance energy transfer. And I understand that previous speakers also have discussed this method and its applications. But now almost 20 years ago, we decided to combine a single molecule FRET with optical tweezers. And this is following the footsteps of many pioneers, Yanagita, Kinoshita, Bloch, Lang, and Lippard. Their goal was to measure conformational changes of biomolecules using FRET, but as a function of applied force. So one of the first things we measured using this combined instrument was DNA-protein interaction. So there's a protein called SSB, single-strand DNA binding protein. And from the structural and biochemical studies, we know that DNA is wrapped around this SSB protein core. And the idea was to use optical tweezers to apply force across the two ends of the DNA, and then use FRET between the green and red dye molecules on the DNA, where you initially should get high FRET due to high proximity, mainly red signal. But as you mechanically unwrap the DNA from the protein's surface, you should see reduction in FRET. So my former student, Robo, who is now at Penn State, one day came to me and then showed me this data. And where as you increase the force in gray as a function of time. We did it 5 times. And you can see really beautifully intercorrelated change in red signal going down and green signal going up. And here the blue is the FRET efficiency, showing that you can indeed actually induce mechanical unwrapping and measure this in real time. And I was so excited, I almost cried because data was so beautiful. But I didn't cry. Instead, I told Rubo to go back to the lab and take some more data. So that became a paper. Now, so why do you care about mechanical forces and their impact on molecular conformations and interactions? So here's a really important work done by the lab of Martin Schwartz, back then in Virginia, but now he's in Yale University. So 2 of his postdocs, Grasov and Hochman, they developed the FRET-based tension sensor inside the cell. So you have fluorescent donor and acceptor and fluorescent protein, cyan and yellow. And then you put a spider silk peptide in between so that you can measure FRET between the two. But if you embed this springy sensing element into the protein matrix inside the cell, then as the cell applies force across the sensor, then the FRET will decrease. And in this particular case, they added a compound that inhibits a force-generating molecule known as myosin, and then therefore force should decrease. And then over time, in just a couple of minutes, they could see a great increase in FRET, because without force generating molecules action, stretching force is reduced, and then you get increased FRET. So it was really exciting to see the data of this type. But the problem was that they didn't know the magnitude of force because there's no real calibration, I mean, experimentally. So this is where we came in. My students, Michael Brenner and Robert Zhou, they use our tweezers and FRET combination to measure FRET as a function of force and show that in both stretching and relaxation cycles, we see highly reversible signature. And the FRET values that they were measuring inside a cell, the force value was about 2.5 piconewton. So that was really our first entry into mechanical forces inside the cell. And so I'm going to tell you things that we have done since, inspired by this collaboration. So our cells have to make a lot of important decisions all the time. And the signals come from chemical and electrical signal in the case of neurons. But in the last 20, 30 years, we came to realize the importance of mechanical signal. So mechanical biology at the single molecule resolution. We know, again, from many work by pioneers in the field, that stem cells and cancer cells can change their behavior quite dramatically based on the mechanical environment. But if you think about it, a little while, then in the end, sensing must occur through proteins that are on the cell membrane that are in direct contact with the mechanical environment. And these proteins are single protein molecules. They must be performing essentially single molecule mechanical measurements on the environment. But there are thousands of these mechanical sensitive receptors on the cell membrane. So collecting single molecule mechanical cues, eventually cells have to make a decision about what to do based on the information at hand. So here's a really nice illustration of the importance of mechanical measurements performed by the cell. So many of our cell types in isolation appear spherical, but if you let them land on a surface, they will often spread out. And in fact, this looks like opening an egg, letting the egg fall onto the frying pan. So you can see cells spread out. And on the left, this is a wild type cell. So it's very happy, nicely spread, looks healthy. But if you actually remove an important mechanical sensing protein, I'll talk more about this, called tailin, then Michael Schi's lab showed that cells actually eventually shrivel and kill itself, undergoing apoptosis, because somehow a cell decide that the surface was not proper enough, then it mechanically and triggers this apoptotic response. So it's really detecting the substrate stiffness is really literally life or death decision for the cell. Now, there are many bonds that have to form between the cell and the substrate to allow the cells to adhere and spread. And these bonds are mainly formed by a membrane protein called integrin. It's a topic of the Lasker Prize, pre-Nobel, like 2 years ago. So let's zoom in. What is inside these adhesion structures? And so this is cell membrane, and outside the cell we have the extracellular matrices. They're forming a basis for our tissues and so on. And the integrins are sitting on the membrane, and it is a heterodimer, alpha and beta heterodimer of many different subtypes. And then inside the cell, in the cytoplasm, there's an actin cytoskeleton that is actually the main source of mechanical force applies through the integrins. And then the integrin, this beta subunit will bind to various adapters that bridge the extracellular matrix through integrins to the actin cytoskeleton. And there are also a bunch of kinases that can sense this and signal for additional changes required. So these are the key molecules that are found in the integrin adhesions. And there are also a lot of multivalent interactions. And once sensing is done, then additional adapters are recruited and additional integrins are recruited to really mature these adhesion structures. So, of course, we like to know how these different proteins interact in terms of binding and dissociation kinetics and so on. But it's not enough to measure this in the test tube, because in the adhesion structures, there's actually physical force being applied across many of these proteins. And it's very likely that their activities and the binding constants will change under tension. So force matters. So you can think about several interesting related questions, force questions. First, let's say you keep increasing the force until you reach a critical force, then what happens? What is the consequence of reaching a critical force? Second, what are the magnitudes of force across many of these single bonds and proteins? And then how do cells sense substrate rigidity? It's an important, decision factor, life and death decision, through single molecule forces, because the stiffness, how rigid the substrate is not a single molecule property, but somehow cells have to use single molecule measurements to decipher the bulk property. So I'm going to address each of these questions using some examples today. All right, so consequences of reaching a critical force. It's known now that force applies through this adaptive protein tailin. I mentioned this earlier. So without this, cells cannot adhere even on a very rigid surface and killing itself. So if tailin experiences high enough force, then it actually unfolds a domain that has a cryptic binding site for another adaptive protein called vinculin. And then vinculin is recruited. And then using additional multivalent interactions, you can recruit additional proteins in the adhesion structure to really grow and mature adhesion structure. And vinculin also binds the actin cytoskeleton. So this understanding was achieved by the lab of Michael Sheets and Julio Fernandez, where they use magnetic tweezers to apply force and use fluorescent labeled vinculin, single molecule imaging, to show that when you stretch tailin above the critical force, the vinculin is recruited, binding to the now exposed cryptic binding site. So that was a very important discovery. What about force magnitudes across single bonds and proteins? So across this vinculin protein, now we know on average force is about 2.5 piconewton in these adhesion structures. In fact, that number came from our collaborative study with Martin Schwartz laboratory using our optical tasers for combination where we calibrated tension sensor that they had developed. And then subsequently, one of the postdocs, Grasov, in his own laboratory, used the same approach to measure forces applied across the tailin protein, and he found it to be about 7 piconewton. And so there was this also very important paper. Then the lab of Tim Springer, my current collaborator, used the same approach to show that across the integrin beta subunit right inside the cell membrane, average force of around 2 piconewton. So you can use this flat-based tension sensor to, actually to deduce force magnitude on average. But one limitation is that you're not measuring single molecules, but Fred is measured from an ensemble of molecules. So this is average of an ensemble of molecules, some of which may not be experiencing the force or may not be even under tension bound to a partner protein. So that's the limitation. What about force between the integrin itself and the ligand embedded in the extracellular matrix? So that is another link, mechanical link between the extracellular matrix, ECM, and the actin cytoskeleton. And for that, there are actually a number of different approaches, including some from my own laboratory, to measure single integrin ligand bound strengths at different force scales. But today I will show you a study that we just published this year based on a new sensor that we called overstretching tension sensor. So I want to come back to a bit of a historic view. So 30 years ago, this important paper was published from Munich, and where they use atomic force microscope to measure the binding force strengths between biotin and avidin. And they show that it is around 160 piconewton, the force needed to unbind biotin from its receptor. And pretty soon, then Evan Evans had a really brilliant insight that saying that actually you cannot really define the adhesion force unless you also specify the time scale at which you are performing the measurements. And that's because, even when you are at a low force, but if you wait there for long enough, then the unbinding can still occur in a mechanically induced manner. So really, if you want to measure the force values accurately, then that you also have to know and measure it under the relevant force loading rate, basically how quickly force increases under the physiological conditions. And you need to know that, and you need to measure the force at the correct loading rate. But the problem has been that for 30 years, we didn't know how to measure the loading rate under physiological conditions. There's another important paper from Carlos Busmantes in 1996. They were able to detect overstretching of long double-stranded DNA. So if you have this 50 kilobase per long DNA, lambda phase DNA, and then you use optical tweezers to apply force, that eventually, one strand will experience enough force so that it can no longer maintain the helical structure, and then DNA will begin to melt, and eventually, above 65 piconewton or 70 piconewton, you create entirely single-stranded DNA. So that's called overstretching transition. And the idea is that, well, if you use a shorter duplex, perhaps this peeling off of one strand of the other can occur at a much lower force. So in the lab, we created long single-strand DNA, 50 kilonucleotides in length, and then we annealed 20 bases long single-strand DNA molecules of 4 different sequences and 4 different GC contents, and each one carries a single dye, green dye, shown here. And then as you will increase the force, and the idea is that DNA with the highest GC content will probably come up at the highest force, so at the last, and so on. In fact, so that's what we can predict. In fact, that's what we see. So these are 4 different molecules bound to the same DNA molecule. And as you increase the force over time, you can see that, first of all, there's a slope, because as you stress the DNA by moving the bead downward, of course, position of its dye will change, more so for the bottommost DNA. But then when the spot disappears due to melting, we have done some controls for photobleaching. So this is mainly due to melting. The first thing that happens is that this lowest GC content comes up. The last one that comes up has the highest GC content. And we do this for many, many times and plot this unbinding force histogram. And then we can see that at room temperature, there's a nice progression in the peak rupture force as you increase the GC content. And now if you replace a DNA strand by PNA that has higher binding strengths, we can even go to close to 100 piconewton before a rupture occurs. So we can cover a fairly large range of force. And we use a loading rate of 0.1 to 6 piconewton per second in this study. And if you measure these rupture forces, or that we call dehybridization force, as a function of loading rate, actually is pretty flat. So as long as the real physiological loading rate is somewhere around a piconewton or so, then whatever we measure here must be an accurate value. Finally, because we perform measurements in living cells at 37 degrees, we also perform calibration at 37 degrees. So now you can go from 16 piconewton to 46 piconewton, depending on the GC content. So now you're ready to use this to look at forces in living cells. So we have this OTS, for overstretching tension sensor, to measure integrin forces. So the idea is that the bottom strand of the DNA has biotin here for surface tethering, and then there's a green dye, and the L is for ligand against the integrin. Then on the top strand, we have the quencher strand as a quencher, so that initially the quencher will quench the fluorescence of the green dye greatly, so there'll be almost no fluorescence signal, even when you have many of them on the surface. But when the cell comes and grabs the ligands using the integrin and applies force through actin cytoskeleton generated forces, then as the force stretches the DNA segment, eventually the other strand will be kicked off, it's peeled off, and then you see a sudden emergence of a green fluorescence signal. So that's the signal that the force has increased beyond this unbinding force. So we wanted to test whether this actually interferes with the cell mechanical response. So we actually put 3 different OTS with different rupture forces, 30 piconewton, 46, 58 piconewton. And the cell is actually spread just fine. So there's no change in cell adhesion. But if you look at the fluorescence signal from these guys, this 30 piconewton OTS actually shows very robust rupture patterns. Whereas if you go to higher threshold sensors, you see more and more reduced. So we can actually see how much force these sensors are experiencing, somewhere in between here, without actually affecting the overall cell adhesion response. So it is good news. We can report on single molecule forces without perturbing integrin receptors function. So I'll show you a fun movie here. So on the left, cell will be adhering, and there's a reflection interference contrast microscopy image of the cell, basically showing the contact area between the cell and the substrate. And then there'll be a purple signal, that our OTS signal being generated as the cells experience force through integrins. And then green will be the increase in OTS between the current frame and the previous frame, just to show the instantaneous force being applied. And then this is a density of forces through some calibration. We can see how many single molecule force events produced per frame in a square micron area. So as you can see in this movie, cells are spreading very nicely, becoming polarized. And then this force is being applied over time. This purple is accumulated signal through the OTS. And then at the tip of this migrating structure, you see instantaneous force building up. And on the right side, we are plotting basically density of this single molecule force events, somewhere in up to tens of molecules per micron square. So we actually have a pretty good sensitivity, and in fact, we can show that we have now a single molecule even resolution. All right, so now we are ready to measure the force loading rate. And so basically, as the integrin applies force through, through this ligand in the surface, then the force will increase. And what is the force loading rate? So for that, we need to measure delta F, force change over delta T, time change. So we need to measure the force across single integrin at least at 2 different time points. So we need a multi-level force sensor, not a single-level force sensor. And we need to measure single molecule force as a function of time. So for that, we made a serial pillar construct. And so here the bottom strand is longer. There's a biotin and the ligand, but on the bottom we have the red and green dyes. And on the top we have strands with quencher that will peel up at 16 piconewton and 30 piconewton. This is at 37 degrees Celsius. So when the force is below that 16 piconewton, everything is very dark. But if the force increases beyond 16 piconewton, then you see dequenching of the red signal. You see red signal appear digitally. And if the force continues to increase beyond 30 piconewton, then subsequently you'll see an appearance of green signal on the same spot on the cell. So that is how we're going to do the measurements. And the next slide, I'm showing you a movie. We are measuring this on a cell. And whenever we see the expected behavior, we use circles to highlight those events. And here are just 3 of these examples. So we synchronize these plots at the moment of red signal appearance when the 16 piconewton sensor, actually gets activated, and then followed by green sensor overlay here. So, for the 3 examples, the green signal is delayed by 5 seconds and 10 seconds and 15 seconds. And we can do this from over a couple hundred molecules and show that indeed the time delay is generally positive. And there's some negative time delay because there are some, various noise sources of various kinds. But main signal is still there. And then by fitting this, you can estimate the average time delay. So that's delta T in force increase. So that to get the force loading rate, we divide delta F, in this case, between 16 and 30 piconewtons, so 14 piconewton, by the time difference. And we get approximately piconewton per second of loading rate for this cell type through this integrin. So that was really exciting because we can finally measure force loading rates in living cells. I'm going to show you just one more data figure here. So we can also do this in immune cells, monocytes, leukocytes, and then it turns out this time delay is much shorter. Instead of 13 seconds, it's 5 seconds. And as a result, the loading rate is not a picodillion per second, but it's 2.6 picodillion per second. So I tell my students that these immune cells, they're maybe rolling in the blood vessels, trying to make some measurements. So they're in a hurry. So therefore, they actually increase the force at a much higher rate. So to summarize, we developed this overstretching tension sensor that has a wide force range, single molecule sensitivity in living cells. I didn't talk about this, but we also refreshed the sensor so that we can measure things in highly dense regions too, and we can measure multi-levels of force. And I think the most exciting message is that we can now measure loading rate in living cells. And I've shown you that we can measure this as a function of cell type, and we can also change the membrane tension and show that higher membrane tension will give you higher loading rate. And it's driven by both actin and myosin, and viscosity didn't have any impact. And in fact, the loading rate we measure between 0.5 and 4 picogillion per second for integrins is actually very close to the typical loading rate for optical tweezers and magnetic tweezers, whereas it's generally much higher than the loading rate used in AFM studies. I also think that these values are likely to be true because in the same year, just a few months later, 2 other labs also use somewhat different approaches to estimate the integrin force loading rate, and they also reported very similar values to what we reported. All right, so I want to then just talk more, discuss not so much about data, but what does this actually mean in terms of mechanosensing? So the third question was, how do cells sense the substrate rigidity through single molecule forces, where rigidity of the substrate is not a single molecule property, but cells have no choice but have to use single molecule forces to make that determination. Because this tailin unfolding and recruitment of vinculin is very critical for maturing the adhesion structures. So the idea that I want to discuss is that the force transmission from the actin flow to extracellular matrix ECMs through integrins and adapters basically have to be used to unfold tailin and for vinculin binding and for adhesion enforcement before the bond between integrin and ligand ruptures. Because the bond has a final lifetime, and before it ruptures, you have to reach high enough force to unfold tailin. So I made these animations to think about it in a more visual way. So we think that the cells can measure the loading rate as a proxy for substrate rigidity. So imagine having a very steep substrate, and then basically ligand is bound to it by a very strong steep spring. And then when ligand binds the integrin, actin flow will apply force. But because of high stiffness, pretty soon the spring will apply very high force through basically when the actin moves, the force will increase very rapidly, and you can reach the force needed to unfold the tailin protein. And then tailin will unfold, and then vinculin is recruited. And even after ligand and integrin bond breaks, you have already done something. You actually have strengthened the adhesion structure. Whereas if the surface is very soft, same displacement induced by the actin flow will not actually increase the force beyond this tailin unfolding force because of low loading rate. And then before you reinforce the adhesion structure, before vinculin recruitment, you basically the bond breaks and you are back to the beginning state and you haven't done anything meaningful. So we think that that's actually how we can translate the rigidity of the substrate through changes in loading rate and to decide what to do. And in fact, a lot of our thinking was influenced by this paper by Kuzaks, although they didn't really think about it in terms of single integrins, more as a collection of integrins in a cluster. Another, I think, interesting set of questions. So far, I have described integrin like passive linker, passive force transmitter. But is that true? And also, what does tailing binding to integrin? Does it actually do something to integrins itself? In fact, in a proposed inside-out signaling, tailing binding can activate integrin. So the first question, I think the answer is definitely no. Instead of a passive transmitter, it is a machine. It is a machine that has really amazingly complex conformational changes, and two main conformations being inactive, shown here, and active. The inactive conformation is populated more than 99% of the time in the basal state, but it has very low ligand affinity. But in the active state, then it has very high ligand affinity. So we wanted to measure single integrin conformational changes. So in collaboration with the lab of Tim Springer, now the same research unit of PCMM, his scientists actually use unnatural amino acids to put two dye molecules on this integrin subunit, a beta subunit, so that in the inactive state you get high fret, and then in the active state you get low fret. And so in fact, from single molecule fret histograms, so in the basal state you see this high fret value. But if you actually add ligands or antibodies that stabilize this active state, then everything shifts to this low fret value for the active integrin conformation. So this type of experiments show that unnatural amino acid constructs and labeling actually they work very well and without perturbing integrin function. So that now we can think about how to answer this following question: does tailing binding activate integrin? In that case, then tailing binding should actually accelerate the transition from the inactive to the active state. But when you measure the lifetime of this inactive state, tailing binding actually did not change anything about the lifetime. So basically, you don't really make activation any faster by having tailing binding inside inside the cell. But tailing binding does increase the lifetime of the active state. Once it becomes activated, tailing can strengthen that a little bit more. Instead, actually, if you measure the lifetime of the inactive state with and without the ligand, so with the ligand, the lifetime is shorter by about a factor of 100, meaning that you actually accelerate the transition from the inactive to active state 100-fold if you have the ligand. So because ligand comes from outside, so it's more like an outside in signaling based on our data. So I want to put together everything that I have told you today into one, slide. So on the cell membrane, integrin is mainly in the inactive state. This is bent state. And then when a ligand binds from the extracellular matrix, it'll trigger a conformational change to the active state. So basically, the induced conformational change outside in, and then tailin will bind, and then it'll link integrin to the cytoskeleton, the actin filament, and through the actin flow, then there will be force applied to the actin through tailin, and this force is resisted by the extracellular matrix in which the ligand is embedded. And if the surface is rigid, then the spring is very stiff, then and you get high loading rate, then you quickly reach a threshold force needed to unfold tailin, and the tailin unfolds, and you recruit vinculin, and then through many multivalent interactions, you then recruit additional proteins to reinforce and strengthen the adhesion structure, and the cell can sit down and lie down nicely and enjoy nice adhesion and spreading. So that is the idea. So I want to just show you this one interesting tidbit about this paper we recently published with Tim Springer. And so Tim managed to begin the introduction of the paper by quoting Merriam-Webster dictionary. Soul is the immaterial essence and animating principle of many, many, many different things. And in the final paragraph of the paper, Tim wrote that watching the integrin machine move using our imaging method showed a different order of structural transitions than suspected from structural similarities. The immaterial essence or animating principle of integrins revealed here might be said to provide a new view into the soul of the integrin machine. So I find this very inspiring. I invite you to think about perhaps using these single molecule real-time measurements to peer into the soul of the molecular machines that you love. All right, so the work on the loading rate was done primarily by Myung-Hyun Jo, a former postdoc who is now a faculty member in South Korea, with the help from Paul Meneses, Olivia Yang, Claudia Karkama, and Sushil Panjani, all graduate students. And then the second, last bit of the work was on integrin conformational measurements done by jointly between Jing Lee, scientists in Tim's lab, and also Myung-Hyun, with the help from Jia Bin-Yan, Taylor Hall, Jun Lee, and Uriel Lopez Sanchez, and Sophia Yan. Thank you very much.
Thank you very much. What a fantastic lecture. Learned a lot. There was one problem that you focused on, and we learned the whole story using the best techniques available for that problem. So I have a couple of questions, but what I'll do is I will open it up. Jyotishman, can you take over? And
yeah, so thank you, Professor Ha. This was very nice. I don't see apparent questions yet. Shudipto, you can start.
Okay. So what I was wondering is that this conformation change of the integrin protein upon binding a ligand is where in the logic does that come in? Suppose we already had that conformation, but it's not bound to a ligand, so it can bind to tallin, but there is no force pulling anything, so tallin remains folded. That would also work, right? I'm trying to get the logic of this conformational change.
Yeah, so that's a good point. So does tallin bind integrin before integrin binds the ligands or after? Right. And I think the data in the literature supports the idea that tallin can bind integrin even before integrin activation or ligand binding.
I see. Okay. And
I think what the idea is that generally, all these proteins have multivalent interactions. So if you have more integrin somewhere and more vinculin, more tallin, they tend to also, hang around there more. So, yeah.
The second question is sort of more systems kind of thing. So we can have complex surfaces which have different features at different length scales, So you can have an alternating hard surface, soft surface, And somehow these integrins, there are lots of them on the membrane. The cell has to decide whether to plunk down and spread and all. Is that integration of signal from different integrins and tallins, etc.? How does that happen? Because the cell never has, in a lab, of course, it has the very nice flat surface and can sit down on. But inside the body, it would have very complex decision to make.
Yeah, so that is a good question. I mean, there are number of ways thinking about this, but maybe I want to mention one paper we published some years ago. We actually wanted to see how many integrins need to experience strong force before cell decides that it's good enough. So for that, we cannot simply reduce the number of ligands on the surface, because if you have very few ligands, then cells cannot spread. You have to have thousands of bonds, right? So what he did was, we had this tension gauge tethers, a DNA, that rupture at high forces, but we put that at very small number, and then we also have thousands of tethers that rupture at low forces. And there we could actually show that cell can spread nicely, even when you have only two strong tethers, as long as you have lots of weak tethers. So the way we think about this is that, if you have two strong bonds, then basically there'll be a line of membrane between the two bonds that can actually facilitate binding of integrin to the ligands. And then even when one of them dissociates, then you can rebind quickly because membrane is still being pulled down. So instead of going far away. And so, yeah, that's a really good question. So what kind of things that cell listen to, right? to make a decision. And again, I think it probably is, still the more local manifestation of this, resulting changes in cell membrane and complexes that will, propagate the information. Yeah.
Great. Any questions further, Jyotishman?
I have a question, Professor Ha. This is just because I'm very ignorant on all the wonderful methods that you are using. My question is, you had these two force sensors, one for 16 piconewton, the other was for 30 piconewton. And because it's a fluorescence kind of a detection, so how many such sensors could you actually use to make a continuous set of measurements of forces? Like, is there a max on that, or you would like to sort of use other techniques to go for a smooth gradation of force sensing?
Yeah, so I think that 3 is clearly doable.
Okay.
And so we're actually working on that. And I think in terms of measurements, 3 is not a problem, right? Because you have, you can measure 3 colors. And I think you can even do 4 colors without real difficulty.
Right.
Generally, the issue is the yield of making the sensor accurately, because even when you 2-color, we actually, on the bottom strand, we have 4 modifications. So we have a ligand and biotin and then 2 dyes. So it was a bit painstaking, because we had to make different pieces and then ligate them and then purify using HPLC. But it can be done. And but if you want to go to 3 colors, then it becomes more complex, and then the yield can go down. And also, I also meant to mention that occasionally we see just sensors with green dye only or red dye only, because even the quenchers can photobleach, we learned.
Absolutely, yeah. Okay, thank you. There is a interesting question from Ajay from JNU. He says, is there any possibility of rise of environmental temperature as a result of this mechanical force? And if so, how can we manage this increase in temperature? This is kind of a Yeah. Did you get the question? It's a
Yeah. So
Yeah.
I guess my guess is that the question is a very philosophical question in the sense that I mean that we are working in water baths, right? Basically, the water cooling is very efficient, so you can I mean, and I think fluorescence excitation actually will generate some heat that get dissipated, but I don't think it will really increase the temperature that much because of efficient water cooling. But maybe the question is more about the temperature in the statistical mechanics sense, right? Because you are changing the entropy of the system. Yeah.
But yeah. Okay. Any other questions, Shudipto? Otherwise, you can take over.
Okay, if there are no further questions, let's thank Professor Tagjip Ha one more time for spending his early morning with us. And sorry about that. I know you have a lecture to give at 9 o'clock, so we won't hold you for very long. But this was really wonderful. I must say this was really wonderful. We went through very slowly from one step to the next, and I think everybody loved it. So thank you, TJ, and we'll see you in a month in Bombay. You, your wife, and your son. So looking forward to that.
Yes,
Okay.
thank you so much for hosting me. It was again real privilege and honor to give this talk, and I really look forward to meeting all of you.
Sure.
Many of you.
Thank you very much, TJ. Thank you.
Okay, bye.
Bye. Okay, I think the day comes to an end. An exciting day, 4 fantastic lectures, and we learned a lot from different aspects of light in biology. There's just the curtain raiser. So there's just the initial flavor of the fantastic series of things that are to come in a month. You see, 4 of these people, there are many more people with new ideas, new techniques that will talk in our OWLS and FCS meetings. Some of them will teach in the FCS workshop. All of these people will talk, and this and many others. I think we have a total of nearly 150 lectures, of course, in parallel. So it's going to be a carnival of ideas. So hopefully you'll see many of you, most of you there in Bombay. And yes, so, and some of you will be going through the online hands-on sessions, which is starting at 10 o'clock tomorrow, Rajatishman. Okay, so where we will show you those people who would not be able to, or many of them, sorry, all of you will be 10 o'clock, right?
10, yes.
10 o'clock, we start again with actual practical lectures about how each technique works. And day after tomorrow would be actually, even tomorrow we will start, I think, there's some demos from the lab. Live demos from the lab. It actually works pretty well. We had to do this during the pandemic, and we are surprised how well it worked because we ourselves were skeptical that it will work. But it actually worked pretty well. Of course, we have smaller groups than this whole 100 plus people, but I think it will be fun. So be there. There are 5 tracks in parallel. I'll give it over to actually Jyotishman, who knows more about the actual organization of this. Jyotishman, just describe what's coming tomorrow or day after.
Yeah. So first of all, thank you for showing so much of enthusiasm for the entire day. I think we had around 150 to 160 people in the first talk, and then, of course, it decreased over the period of the day. But thank you for still continuing to join into the Zoom account as well as watching through YouTube. It was pretty successful, and Shudipto, thank you very much for organizing this. It was all
Hey,
Shudipto
Jyotishman, actually we cannot hear you very well.
Hello, can you hear me? Can you
Might
hear
be
me?
better.
Yeah.
Just continue.
Okay. Yeah. So I would like to mention that we have to thank Shudipto Maiti for putting this program today, the entire session with 4 speakers of pioneer stocks. So thank you, Shudipto. And tomorrow we start with the actual online teaching, which will be starting at 10 a.m. with 5 tracks. There will be common talks all in the morning and a bit in the afternoon, from FCS to ultrafast to Raman to AFM force and then to single molecule. And these common talks will be available to everyone. And then we will move into the individual tracks with people going in, the teachers going in a little bit more technical details, showing analysis as well as demonstrating the equipment. And that would continue on Sunday again from 10 a.m. So we look forward to your continued participation. Each of these tracks, since they're online, you can jump over and see what's going on. In each of these tracks, we will have breakout rooms and you can go from one breakout room to another. But it would be good if you maintain in one track, because some of these things will be asked to you as pop quizzes in the actual in-person teaching. So please do maintain a notes. These are basics and then a little bit more to it through technical sessions. So thank you very much for joining today. And Anandada, you want to say something more? Anandada? Hello? Okay.
He must have just left or something.
Okay.
For the meeting.
Okay.
But I
Yes.
think we're done, right?
Yes.
Maybe
Yes.
Devanjan wants to say something. I can see him unmuting himself. Devanjan, you want to say
No,
something?
I'll just call Jyotish Banda. I have some technical issues.
Okay.
Oh,
all
I
right.
just wanted
Okay.
to know that. Yeah.
Sure.
Okay, that's okay. So we finish this session, end this session, Jyotishman?
Yes. Thank you very much. We can finish.
See you
this.
guys
Yes.
tomorrow.
See you guys tomorrow. Thank you.
Bye.